and therefore I am privileged to be its founder chairman. So for last 36 years, with the active association of very senior scientists from India and abroad, we had been running this society and the journal. And as you can witness, that it is showing an exponential growth throughout the world. And its journal is one of the most loved journals of plant sciences as on today. Since 2019, the journal is being co-published by Springer Nature, and ever since we have attained new milestones in various fields. As on today, we are having the editors, more than 100 from nearly 47 countries, who are dedicated for processing various manuscripts assigned to them in different areas. We are having the senior editorial board members and the subject editors who take up the papers for processing further. Of course, we encounter some problems pertaining to the editorial process and to the reviewing process, because most of the scientists, they are in active service. And particularly those scientists, they are facing paucity of time because they are overburdened with their own assignments in their universities or in their institutions. It is the major cause that our handling editors and our senior editors, they are unable to complete the process of reviewing in time. That is the biggest drawback. And this drawback can be overcome only when our reviewers take a little interest, or we identify those editors and those reviewers who can work as a core group of the society. I am emphasizing on the core group because we have to have a core group of reviewers and editors who can work tirelessly for the journal. The casual approach of anybody will harm, because if any part of the society indicates slackness, the authors, they are not going to spare us, because they keep on reminding right from 15 days onward. Sir, what is the status of our paper? Why it is editor assigned? Why it is with the handling editor? Why the reviewers are not there? Why it is so much delayed? We submitted it 15 days back. There is no process on the editorial board manager. So you will not believe that almost 4 or 5 hours in a day I spent in responding to their emails and their queries. Sometimes after 1 month of their submission, they virtually threat that we are going to withdraw this paper from the journal. I think there is a difference. I am taking just 2 minutes before we start with the presentations. There are two different thoughts. One thing, that without going for peer review, without seeking the expert opinion of the reviewers and the editors, we should accept the paper and publish it as such. This is the most suicidal act of the journal, and the journal will go to the graveyard. And nobody, even those people who have opted for publishing it quickly, even those people, they will not contribute. So this option, we are not prepared to go. The second option is that we should focus on the quality of papers which we are receiving. And for quality of paper, we have to seek the opinion of our expert reviewers and the editors as well. Of course, when they invite the reviewers, the reports come and the editor, they go through the report, that what type of report they have submitted. If they are not satisfied, they send it to another reviewer. And unless and until the editors are satisfied, they do not accept it. Because I am the last step. I am the first step in sending the paper to the editors, and I am the last step to submit the final decision. So in between, there are so many channels. And because of API and because of so many other factors, I receive mails that my student is waiting eagerly for the acceptance because thesis submission is due. I am waiting because my promotion interview is due. I am waiting for the acceptance because this and that and so and so and so. Thousands of questions every day we are dealing with. So if the chief editor office is busy with all these affairs, I think that is cumbersome, and we cannot focus on the quality of the papers. So this is one thing which I explained, because all of our esteemed fellow awardees, they are going to be a very important part of the society, either in processing the papers or as editors later on, or sometimes in the future, or they will be neck to neck with us. They will help shoulder to shoulder with the editorial board to make this journal the most loved journals in the world. So this is one aspect. For selecting fellows, because we are having three different categories of awards. One is the fellow award, another are young scientist award in different areas, and the third one are senior scientist award, which is provided to almost very senior faculty members. Of course, I am very much happy that for fellow awards, very much senior people, very much senior scientists like Dr. Sanket Joshi, I can see, they have submitted their nomination. So this is their trust and love toward the society, because they trust and understand that the process of selecting the awards by the Society for Plant Research is absolutely a transparent exercise. No part and prejudice, no biasness, nothing. It go by merit. And you can see that yesterday we also had a very good lecture series of fellow awardees, 9 fellow awardees, they delivered their lectures. And today also we shall be having 11 lectures. So our executive body frame a screening committee headed by the chairman of the screening committee. This year, Professor D.K. Srivastava, Director Extension from Himachal, was the chairman of the screening committee, and 7 members from different parts of the country were there. And for just 20 awards, we received more than 100 nominations. It is the love and affection of the scientists who have great trust on the society and its awards and other activities. So the entire screening committee screened the nominations, and by virtue of the merit, throughout merit or overall merit, they decided that these scientists should be selected. So this is one very important step, because some of the societies, they just hand over the awards just without going for any formality. But our executive understood that we should have their presentations, because at least all of our colleagues should know that the decision made by the screening committee is perfectly worth. And let us know that what our fellows are doing, what their laboratories are doing, and in what way they can be associated with the activities of the society. So that is very important. Otherwise, giving award and receiving award is one formality. So by or through awards, we should understand each other and should know that what type of research we can collaborate, what type of research we are doing. So at the outset, let me congratulate the fellow awardees in this session on behalf of myself as founder chairman and secretary general, and on behalf of our president, Professor Vandana Bose, who, despite of her other engagements is present over here. Professor Abhay Pandey, I can see. The editor-in-chief who is working shoulder to shoulder with me as associate editor-in-chief with Vegetals, a very prominent scientist from Allahabad University, and all other colleagues are here. So I extend my heartiest welcome to you all and congratulations. So with this, we can start with the first lecture with the permission of Professor Vandana Bose, our president. Can we start proceedings? So thank you very much, and I would request all our attendees and awardees to kindly keep their mics mute. And our first lecture will be started within few seconds.
Good afternoon. This is Sanket. Shall I start?
Yeah, Professor Sanket Joshi, please start.
Okay. Thank you. So I hope my slides are visible to you, and I'm audible.
Yeah, perfectly. Perfect.
Okay, perfect. Thank you very much. So thank you, Professor Vatnagar, Society for Plant Research Committee, Vegetos, and all the members who thought my work is worthy of joining SPR as a fellow. So thank you very much. So time is an essence, and we all have limited time. So I would like to finish my speech in a given time limit, which is around 10 to 12 minutes. So without going much into it, so I'll be mainly talking about microbial biotechnology. What are the different kinds of innovations in energy and environmental sustainability me and my group is doing since past 21 years or something. So basically what I will cover is the concept of One Health and some of the unorthodox environmentally friendly microbial applications in energy field and how to take it from lab to the market, not just keeping it inside the research laboratories. And what are these kind of microbes and biosurfactant applications? What are the different challenges and what are the future scopes? What we all can do into it. So as an Indian, it's not a new thing. We all know Vasudhaiva Kutumbakam, which is the world is one family. This is what we are believing in and living in India since past centuries or something, and which was basically when we hosted G20 in 2023. This was the motto of our country. And nowadays it's coming from Western world and other scientific societies also, which is known as a One Health. So what they are trying to tell is what we are living since past so many centuries, like environment, societies, host, and our microbiome, which is inside us, they all are interconnected. Nothing is separate. So we all are living in a one world. So basically in One Health, it follows a simple guiding principle, like to protect the health of human population, because of course we are humans and we are defining these things. So we are putting forward like more human-centric definition. So it is essential to protect human population, to preserve the health of animal populations as well as the ecosystem. So it's a kind of a Venn diagram where you can see that we are no separate entity as compared to animal health or environmental health, and it is all interconnected with human health. So our group is working on different aspects, but today I will be mainly focusing on different kinds of human activities, which is the concern of contamination of environment and by releasing toxic substances and everything, and how it is connected to energy field. So I'll be discussing part of my research work about offbeat environment-saving applications in energy or petroleum industry, what we are serving industry since past 20 years. So we do work on enhancing oil recovery, solar power, and many other things. But here we will be mainly talking about some of the environment-saving technologies, like where these kind of heavy crude pipeline transport it can be used, oil field souring and sulfate-reducing bacteria, biopolymer, polymer degradation, remediation of some of the oil contaminated sites, produce water issue is one of the very big issue all over the world, and some of the next generation fuel, which is natural gas hydrates, and how to do carbon capture and sequestration. So some of the topics I'll be touching briefly. So to start with, first is application of biosurfactant to save money as well as the environment for doing the crude oil transport by pipeline. So before we go into biosurfactants, let's understand how biosurfactants are different from surfactants. So we all know surfactants. They are comparatively smaller molecule. We call it as a detergent, which we use in our day-to-day life. It's a smaller but quite versatile molecule. It has both hydrophilic as well as hydrophobic moieties, because of which it is having affinity for oil as well as water also. So what does it do basically is it reduces the tension at the interface of air and water or oil and water. And by doing so, it does mixing of oil and water and removing. So biosurfactants are nothing different. They are basically biological based surfactants which are produced by microbes, by plants, as well as animals. Like we also produce surfactants, like which is known as a lung surfactant. So it protects us from infection and everything. It is produced in our lungs. We can use plant-based biosurfactant, but it takes some time to grow plants and everything. Microbes is comparatively easy to manipulate as well as grow into the fermenter in a lab and everything. So we basically go for bio-based surfactants from microbes. So how it can help in crude oil transport? So if you compare, if you know the history of crude oil, most of the crude oil resources are very heavy crude oil now. So when you say heavy crude oil, it's very heavy. It's like a Shrikant, what we say. So viscosity of water is around 1 centipoise. So when you say heavy crude oil, we are talking about viscosities in the range of 1,000 times to 10,000 times more viscous. So you can imagine it is very viscous and it is very difficult to transport crude oil from the oil reservoir to the oil refinery. So if you compare how much does it cost to transfer the oil from oil well or oil reservoir to refinery, it will cost around $20 if you do it by trucks. It will cost roughly $10 to $15 per barrel if you do it by rail, or if you do it by pipeline, it will cost you around $5. So it is comparatively cheaper if you do it by pipeline. In terms of safety comparisons, if you see how many number of fatalities per year can go, this is an average value. It can be plus or minus. So you can imagine more accidents can happen if you are doing it by road. So around 10 to 12 people can die per year on an average if you do it by road transport. Same goes for railway, roughly 2 to 3 people can pass away. But if you do it by pipeline, normally less labor is involved into pipeline until unless it's a maintenance or a blast or something. So less number of people dies by pipeline transport. So overall, pipeline transport is much more beneficial. How does it work in industry? You go by reducing the viscosity of your crude oil. So if the oil is less viscous, it's easier to transport. You can reduce the friction. You can do in situ upgrading. So we are focusing mainly on emulsification, like making emulsion of oil in water. So by doing so, you can reduce the viscosity. For this, we have selected different kinds of oils. This is the oil which was very heavy. You can see the viscosity is around 28,000 times more than water. So we mixed it with our biosurfactant, and we managed to get viscosity reduction in the range of 98 to 100%. So it's like water. And by doing so, we reduce the cost of around 36% reduction in the pump and electricity requirement. You need electricity requirement to transport oil from one point to another. So we managed to save a lot of energy as well as money saving technology, and biosurfactants are biodegradable. So this was one of the applications. We also worked on souring of oil well. Souring is something similar. When you say doodh khatta ho jana, when your milk gets spoiled, you say milk is sour. Same way, entire oil well can also get sour. It's mainly because of sulfate-reducing bacteria. It is quite poisonous gas. It is going to make a lot of issues in your pipeline. It can lead to workers' death also. And it's all because of sulfate-reducing bacteria. In this slide, it shows what are the different ways by which you need to be careful when you are working in oil well. But I like to see it as what are the different ways by which you can die. You can die by respirable silica sands while drilling the oil well. You can do by vehicle accidents. You can go by explosions, as well as something is similar to chemical exposure. So hydrogen sulfide is one of the biggest reason by which people can die. Around 700 ppm. Plus, if you smell hydrogen sulfide, you can die with a single breath. And it's a highly poisonous as well as explosive gas. Some of the oil wells where we worked was having around 50,000 plus ppm hydrogen sulfide. It can lead to so many casualties. These kind of warning signs are necessary. Camels can die, cattle can die. And same thing can happen in the natural gas transport also because of hydrogen sulfide. So our group was working mainly on providing different kinds of solutions to prevent hydrogen sulfide production, as well as provide the mitigation of souring of oil well, so that we mapped around 180 plus oil wells in Oman, and this is what we are trying to do here with ONGC also. This is another application by which you can prevent the accidents as well as environment. Another application is oil field produced water. So normally oil and gas industry is one of the biggest industry where you need a lot of water. To produce oil, you need a lot of water. So when you produce water with oil, it is known as produced water. You separate the oil. As you can see the picture on the right hand side, The amount of oil is very less when you produce it, but amount of water which comes out of oil well is very high. How high? Per barrel of oil, roughly you produce around 9 barrel of water. And if you look at the quantity of amount of water produced worldwide, Oman is given an example here because we work with Oman mainly. Data is given for US and all the other countries. So we are producing around 900,000 cubic meter of water per day. Not liter, cubic meter. It's a huge quantity of water which comes out of oil well. And what is the kind of water we are talking about? It's nightmare of any water treatment company. It has everything what we don't want in our water. It has very high salinity, crude oil, traces of naturally occurring radioactive material, TDS, heavy metals, chemicals, everything. And to icing on the cake is most of the part in the world, we don't have a lot of water. So this is one of the biggest challenge oil companies are producing. To produce oil, you need more water. And when you use more water, you end up producing more produced water, which is very toxic. So they normally do percolation ponds, open percolation evaporation ponds, because they have a lot of land in Middle East. We don't have it here. Or else they do subsurface injection. They re-inject the water back into the well. This might contaminate the subsurface water part and everything. So they are looking forward for some solutions because the amount of water which comes out is huge. It's more than 1 million cubic meter per day. So we provided them water to value program using nanotechnology as well as by using algal technology. So if you use forward osmosis using nano-treated membrane, so you can get very good quality of water, but it's quite expensive. Or else you can do the treatment by algae and you can generate biofuels. So you can go for biodiesel or you can go for hydrothermal liquefaction. And if the quality of water is good, you can also produce some high value fertilizer as well as chemicals. Algae, I won't go into detail. I think you all know algae much more better than me, but algae can grow in fresh water as well as in a very high saline water as well. These are the market comparisons. Different kinds of products are available in the market in the range of $11 billion per $1.8 trillion dollars per year. So different applications are there for algae. This is a bit older data. So I think the current data is even much better. So we did these kind of treatment of produced water from oil wells, and then what to do with the treated water. So we don't have water in most of the places. So this was a case study. You all know jatropha. They tried growing jatropha. They tried growing cotton plants as well as different kinds of cash crops. And same way you can do carbon capture and sequestration. You can get the water for agriculture land as well as some of the water can be taken back for oil recovery. So this was the solution we gave to them for reuse, treatment, and entire cycle for the oil well treated water. You can have biochar, you can have biocrude, as well as clean water for discharge. And the last application I would like to talk about is how biosurfactants can be used for natural gas hydrates. So what is natural gas hydrates? You can see in the right-hand side picture, it's a crystalline compound, which is basically a gas molecule bound between the water molecules. So between the hydrogen bounded lattice, you will have the molecule. And gas hydrate deposits are considered as a future fuel. We have a lot of gas hydrates, a lot of research is going on in Godavari Basin, IIT Chennai, a lot of group is working on it. Some group are working on collaborative research with Japan and other places also. So if you apply a fire to these kind of gas hydrates, then you can see the burning ice kind of a thing is there. So if you keep it on your hand, it will burn and only leave water as a traces of water. So this is kind of a burning ice or gas hydrates. So there is a fantastic chemistry behind it. Gas hydrates are normally made up of methane. So it's a methane gas hydrate. So if you push CO2 in these kind of methane hydrates under subsea bed, then the CO2 can be preserved there and this methane can be used as a fuel. So you can do both the things. You can release methane, you can use it as a fuel. At the same time, you can preserve CO2 under subsea beds or in the different kinds of deposits. So to do this, hydrates can be formed using different kinds of promoters. These promoters are chemical surfactants. So we said, why don't we use biosurfactants instead of chemical surfactants? Because they are biodegradable and environmental friendly. So we are working on these kind of biosurfactants, which will help not just in generation of gas hydrates, but also helping in carbon capture as well as sequestration. So this is also another kind of research we are working on. Some of the publications, of course, being into academics and everything, when you are doing research, you need to publish papers. Our group is actively involved wherever it says microbial technology, be it science, be it engineering, be it environment, space, anything and everything. These are some of the students working with me in the lab with fermenters. We are also working on different kinds of other applications, enhancing the oil recovery, transport, which I have shown, sovereign control, We are also working with natural gas transport, biofuels, biodiesel and bioethanol, third generation, diagnostics and microbial products we are working on from lab to industrial scale. Plant-based products also we are working on for probiotics and other things. So just to summarize quickly, because of the paucity of time, so there are tremendous potential and scopes are there for being a microbiologist or a biotechnologist. Either you can do a job or you can also be an entrepreneur. We recently started a company for doing this work in the field of energy and environment. There are certain challenges and future prospects. We need to keep on learning new, and the generation and the time is for interdisciplinary technologies. I can't be an expert of only one thing. I need to know other departments, other fields, as well as I should have a connections and collaborations also. I should be open to learning new things, and we can't ignore the potential of artificial intelligence and machine learning. We are using it widely in simulations and different kinds of models for generations of these things. We have to learn it. There is a lot of work to be done, and applications in energy industry and pharma sector and diagnostics market is another multi-billion-dollar industry which can be properly harvested, and there is enough opportunity for all of us. So would like to acknowledge research funds and facility provided by Amity and collaborators and different kinds of funding agencies, members from Oman, India, from Ulster, Kyushu, Japan, Calgary, Kazan Federal University, Russia, RJIPT, Amity, UPS, Dehradun, and some of the oil companies, Schlumberger, SNF, France, for giving us access to softwares and high-speed computing, and of course, 40 plus students, without which it won't be possible for me to sit here and discuss about different kinds of work. So definitely big thanks goes to all the students who have contributed over the years. And with this, I would like to finish. I took 12 seconds extra, so apologies for it. And feel free to reach me. This is a beautiful campus, Amity University, Rajasthan. So whenever you come to Jaipur, please do let me know. I would be more than happy to see you and discuss more about other work, what we are doing here. So thank you again for giving me this opportunity.
Thank you, Professor Sanket, for this brainstorming lecture. I hope everybody enjoyed. We are not having the question session here because of the paucity of time, as you know.
I understand.
But everybody is open to reach to the scientist concerned or to us, which will be diverted directly to the scientist. And we shall be having a 15-minute session after this complete session is over for question and answer. So kindly stay with us till then. And now I would request Dr. Deepu Vijayan from Botanical Survey of India, Scientist D, to kindly give his presentation. Let me congratulate at the outset, and we are eagerly looking for your presentation. Dr. Deepu, please. When the slides of Dr. Deepu Vijayan reaches to our office, and let me also request all our esteemed fellows and attendees to kindly keep communicating your very good quality researches to the journal Vegetos. And also keep on citing the researches which are published in the journal during last 36 years. I will not say that age-old references should be quoted, but at least for last 4, 5 years or 6 years, whatever researches have been published in the journal, kindly keep on citing, because the scenario of different agencies, they keep on changing and they are diverting from one parameter to another. So citation is one important parameter other than the self-citation. And you know, as on today, the journal Vegetos is having 67% rejection rate. I should start from the negative point of it. So the rejection rate is not because we are happy with it, but because the practice of submitting review articles by the amateur scientist, by the scientists who are not even on the regular payroll, who have just started their career, those who do not know the subject even at the basic level or at the foundation level, they are writing review articles. So that keeps us in a very odd situation what to do with it, because artificial intelligence is coming up. So it is helping writing many papers like this. Are you okay, Dr. Tipu, now?
Yes, sir. Yes, sir.
Okay, you can do or our office will do?
Sir, I'm unable to move the slides.
Okay.
So I think.
Yes, our office is doing. Please kindly continue.
Yes, sir. Sorry for the technological inconvenience.
Not at all. Please continue.
Yes. Good afternoon, all. Next slide, please. As you all know, Northeast India, it is among the 36 identified global biodiversity hotspots. Indo-Burma and Himalayas lies in this region, and it comprises of 8 Indian states. And this is region is covered by the mighty Brahmaputra also, Brahmaputra river system and its tributaries. And one more highlighting thing is the highest rainfall, averaging around 10,000 millimeters. Next slide. And this Northeast India covers, the Northeast India with having 220 ethnic groups and equal number of dialects makes it a hugely diverse region. And it is unique for its culture, handicrafts, martial arts, scenic beauty, and different kind of forest. Next slide, please. And regarding the plant diversity status, it forms the richest reservoir of floristic diversity and supports about 50% of total 18,000 flowering plant species of India. And interestingly, 825 orchids out of 1145 orchids is reported from here. And the same is the case with the rhododendrons, bamboo, and canes as well. Next slide, please. The highest plant diversity is reported from the states of Arunachal Pradesh, followed by Sikkim. And according to Red Data book published by BSI, 10% of flowering plants in the country are endangered. Of approximately 800 are reported from Northeast India. Next slide, please. And there are some famous national parks. This is situated in Northeast India. Like, for example, Kaziranga, Manas, and Nokrek National Park. Next slide, please. These are some of the wildlife sanctuaries. Next slide, please. And now let us see what are the current challenges in biodiversity conservation. The first one is the habitat loss and fragmentation, followed by climate change, invasive species, pollution, overexploitation, and disease and pathogens. Next slide, please. This photo was taken from Arunachal Pradesh. You can see the cutting of trees for road widening, and you can also see some rhododendron plants, young rhododendron plants in this picture. I will explain about this in my coming slides. Next slide, please. So there are some questions arises. So what are to be conserved? What is the immediate step taken? What species are endangered? And what is the general distribution of these species? Is the plant species, subspecies, or a taxon are rarely produced hybrid, polyploid, or mutant? Is the species is truly endangered or likely to be endangered? Next slide. And why to be conserved? Because what we don't know what we are losing. We don't know what we will use. Next slide, please. And next question is where to be conserved. Next slide, please. And the final question is how to be conserved. The best is the in situ conservation, and the very next is the ex situ conservation. Next slide, please. These are some of the ex situ conservation methods. Next slide. These are the methods of germplasm conservation. Next slide, please. It is continuing. Next slide, please. Next slide. So keeping in view of all these challenges, we have proposed for some projects, both internal and external funding, mainly focused on the endangered and endemic plants of Northeast India. And we have completed one DBT project. There we have focused on two plants. One is Rhododendron vatae and another is Rhododendron macabinum. This was collected from Zuko Valley, Nagaland. It is a hub of rhododendrons. Next slide. The fourth picture shows the picture of photo of Rhododendron vatae. There is only one big tree is remaining in this particular valley. So we could collect both Rhododendron vatae and macabinum from that side. Next slide, please. Next slide. We could successfully establish a in vitro micropropagation protocol for both the plants. Next slide, please. And we have carried out the genetic integrity analysis before reintroducing into the natural habitat. Next slide, please. And the main objective of any tissue culture program is reintroduction. And we have successfully reintroduced these two plants into the natural habitat of Kisama, the Naga heritage village in Nagaland. About more than 100 plants were reintroduced in that particular area. Next slide, please. Next slide. These are some of the publications. Next slide. Next slide. And we have conducted another project which focused on the micropropagation and screening of secondary metabolites of some medicinal orchids in Meghalaya. These are some of the orchids selected for the project. Next slide, please. And we have successfully standardized the micropropagation protocol for, next slide, for medicinal orchids using organic additives, especially banana pulp and coconut water. Next slide, please. These are the publications from the project. Next slide, please. Next slide. And we could successfully bring out an electronic book named Micropropagation and Screening based on polyphenol and antioxidant potential of 6 medicinal orchids in Meghalaya last year in 2024. Next slide, please. And this is Pioneeria khasiana. This is an endemic plant to eastern Himalayas. And it was this particular plant was collected from North Lakhimpur, Assam. Next slide, please. And we could successfully reintroduce these particular in vitro raised plants into the natural habitat in Meghalaya as well as in Assam. Next slide, please. These are the publications. Another project is we focused on the endemic and threatened plants of Northeast India. There we selected Rhododendron formosum and Rhododendron innocuole and Adenandra griffithii. All these plants are endemic to Northeast India. Next slide, please. And we have successfully reintroduced this plant in East Khasi Hills, Meghalaya in 2023. Next slide, please. Next slide. Again, reintroduction was carried out in Latmasiang village there in Meghalaya in 2023. Next slide, please. Adenandra griffithii also, we did the reintroduction, but unfortunately, we could not do any reintroduction regarding the Pioneeria barringtonifolia. Next year, we are planning to do that. Next slide, please. These are the cultures still maintained in our tissue culture laboratory. Next slide. These are the publications. Next slide, please. Next slide. We have conducted another important project on taxonomic revision and phylogenetic study of Zingiberaceae. Since Northeast is a hub of Zingibers. Next slide, please. These are some of the interesting Zingibers collected during the period of study. Next slide, please. Next slide. And regarding this particular publication, once we published this particular paper, the local newspaper in Meghalaya, they emphasized on the conservation efforts of BSI for conserving this particular plant species. Next slide, please. We have another successfully completed another project on NMHS project on conservation of threatened plants in Indian Himalayan region. There we could standardize some protocol for the phytochemical evaluation of some medicinal plants in Northeast India. That is like untouched. It was an untouched area. Next slide, please. Next slide, please. Next slide. We have come across an interesting plant that is Indigera glandiflora. It was collected from Nagaland. And the folks of Kimangwan tribe, Nagaland, say that the wounded tigers heal themselves by eating the leaves of this particular plant. So we could publish this particular plant species in the Vegetus journal itself. And so many people appreciated about this particular study. Next slide, please. Next slide. We could focus on an endemic genus Hypericum of Northeast India, especially regarding the chemical composition and antioxidant activities. Next slide, please. These are some of the interesting Hypericum from Meghalaya. Next slide, please. And we could recollect an endemic plant Hypericum lobi after 133 years of type collection. And we have a new addition of Hypericum williamsi to the flora of Meghalaya, Assam, and Nagaland. Next slide, please. Next slide. This particular plant, Rhododendron inequal, due to the state highway of construction of Shillong to Dauki, this particular species was about to die. So we could successfully relocate these particular plants from that area to our garden. And fortunately, all the plants survived and it was in flowering. Next slide, please. These are some of the executive propagation of seedlings. Next slide, please. This some macro propagation, and we have focused on 47 indigenous plants, and we have successfully raised approximate number of more than 1 lakh seedlings, and more than 87,000 were distributed to various stakeholders, villages, universities, and colleges. Next slide, please. These are some of the photographs. Next slide, please. Next slide. Next slide, please. These are some of the activities what we have conducted throughout Northeast India. Next slide, please. Some awareness program and capacity building and conservation efforts. Next slide, please. This is the awareness and plantation program at Sora, Meghalaya. Next slide. This is at Sora and Nongpu, Meghalaya. Next slide, please. This is at Kohima, Nagaland. Next slide, please. This is at Senapati, Manipur. Next slide, please. And we have conducted some hands-on trainings in GIS mapping and database management, especially of ENM of plants. Next slide, please. And we have an eco-friendly approach to mainly focused on the mission life activities, reduce, reuse, and recycle. We use jute bags instead of poly bags in our nursery. Next slide, please. This is our poly house. Next slide, please. We have an experimental garden in Barapani where we conserve most of the endemic and endangered plants. This is the National Orchid Area. Next slide, please. So some of the publications. Next slide. And the scientists, we have identified around 1.7 million species on Earth, but there are still identified and many are disappearing every day. So there are some questions. Did some of the vanished species hold ingredients we could use for medical cures, or did some provide home or food for other species which may starve without them? So we are only beginning to understand how complicated our living relationships are. Next slide, please. So it is our duty of today's generation to preserve this nature and its resources for the generations of tomorrow. Next slide, please. And I would like to thank my director, Dr. Ashigo Emao, my head of office, Audio sir, and my students, Dhavanri, Suparna, and Larima. Thank you very much.
Thank you very much, Dr. Deepu, for wonderful, colorful slides. And we all felt that we are traveling somewhere in Meghalaya and Shillong.
Thank you, sir.
It's so beautiful, and the conservation of different plants is appreciable.
Thank you, sir.
Thank you very much, and congratulations once again for being nominated for this prestigious award of Fellow of Society for Plant Research.
Thank you very much. Sir.
Mm.
Thank you very much, sir.
Now I would call upon Dr. Anayatullah Siddiqui. Dr. Anayatullah Siddiqui. I think he was requesting me yesterday because we were short of time yesterday. We could not accommodate his lecture. He might be traveling anyway. In case he joins, we will come back with him. Dr. Arpita Srivastava. Dr. Arpita Srivastava.
Yes, sir. Yes,
Yeah.
sir. Yes, sir. I'm there, sir.
She is scientist in Division of Vegetable Science, Indian Agriculture Research Institute, New Delhi. So, Dr. Arpita, let
Yes,
me
sir.
congratulate you at the outset for being nominated for prestigious Fellow Award of Society for Plant
Thank you so much,
Research.
sir.
And I am sure that you will join hands in providing good progress to the journal and the society. So kindly
Definitely,
continue with
I'll
your
try my
presentation.
best, sir.
Kindly continue with your presentation.
My slides are visible, sir?
Yeah.
Okay. A very good afternoon to all of you. I am Dr. Arpita Srivastava, Senior Scientist working at Indian Agricultural Research Institute. So first, I would like to thank the Society of Plant Research for conferring me this prestigious fellow award for my short period of research what I have done. So I'll just brief you all today. So what all work we have done at IRI, especially with respect to the chili crop. So the title of my presentation is Turning Adversity into Advantage. So as we go through the slides, you will be understanding why I'm talking about adversity into advantage. So just briefing my journey, I did my BSc from Vishwa Bharati, West Bengal in 2007 and MSc and PhD from GKVK University of Agricultural Sciences, Bangalore in the subject genetics and plant breeding. So I joined ARS in July 2012, where my first posting was at IRI New Delhi, and I am continuing here only in the Division of Vegetable Science. So when I was posted at Division of Vegetable Science, and I was allotted the chili crop from the director of IRI, because chili was a closed crop there, and because chili was my PhD crop also, so he had some hopes whether I can restart the research on chili crop there. So the major reason why chili was stopped there is citing the adverse condition in Delhi. So talking about the adverse conditions, the temperatures are quite extreme, both very hot summers and very cold winters. And one of the disease, leaf curl infestation, which is caused by begomovirus, that was is very severe in nature here. And because of the fluctuation in environment, the breeding period which we get at Delhi is also very short. So because of these reasons, maybe the chili program was closed at IRI. So I took up the challenge to just restart the program. So I did some basic interventions in the crop production technologies, which were not new technologies, which were already known, developed by various scientists. So I did some permutation and combination to start so that a healthy crop can be taken up at New Delhi station. Because for any crop to be evaluated, the horticultural traits, the evaluation of horticultural traits is very important. So for that, like tray sowing in pro trays, or sowing in only pro trays is compulsory. Then taking up chili cultivation under low tunnel condition at Delhi, especially during the winters. And some adjustment I did in the sowing time and all, so that we can synchronize the most critical stage of the crop growth, that is the reproductive stage, during which we carry out all the crossing and hybridization programs and so on. So my focus area for this was basically three focus area I started with. First one was the leaf curl disease, which was causing a havoc in this region. Then second in my mind was that I should have some high temperature tolerance and cold temperature tolerance line, because they are very important, especially for the farmers of this North Indian plain zones. So, coming first to the story of the leaf curl resistance breeding, we started with the identification of resistant genotypes, then followed by identification of major viruses causing this disease, then understanding whether the resistance in these resistance sources are against the vector or the viruses, then whether the plant age has any influence on the resistance, then genetics, transcriptomic analysis of the resistant and susceptible genotypes, and finally we have also done some initial mapping studies for leaf curl resistance in Chile. So initially, when I started my work, it was some 65 germplasm lime I had assessed for 3 continuous years, where 3 lines DLS cell 10, WBC cell 5, and PBC 142 I found resistant to leaf curl. And till now, whatever research I have done, at present I have 18 genotypes as well as advanced breeding lines which show resistance to leaf curl disease. So based on all the screening work we had done, we have got some good publications here. Then the next step was for the identification of the predominant viruses which cause leaf curl at New Delhi. So doing based on the virus specific screening of the infested samples at New Delhi station, we found chili leaf curl virus to be the most predominant one, followed by tomato leaf curl New Delhi virus, tomato leaf curl Jayadevpur virus. These are some gel pictures of the virus specific screening. Then our next aim was to understand whether the resistance in the genotype was against the virus or the vector. Vector here is the white fly. So for understanding the resistance against the vector, we used both the free choice method and the known choice method. And in this, through both the method, we found that in the clip no choice method, we had used some clip-on cage method. We were forcing the white flies to feed on the genotypes and give their and we did the studies, and studies was basically done in the form of studying the number of nymphal stages, how many nymphs were being produced by the white flies, what was their egg density, how long did the white flies take the developing on the genotype, their oviposition rate. And in just we concluded that DLS cell time 1 of 10, one of our resistant genotype, it was found to show resistance to the white flies because it were manifested very less number of egg hatching on the on this genotype, and the white fly in general took more time to complete its life cycle on this genotype. So DLS cell 10 was found to show some amount of vector resistance. Then we try to understand the resistance against the viruses. So these are some of the pictures of the field evaluation which we have done, where we can find the susceptible genotype just behind the resistant genotype. And we tried to measure the viral titer load. And we found that our resistant line had very low viral titer load. It was not devoid, but it showed low viral titer levels. And later on, we found that it is basically a symptomless carrier of chili leaf curl virus. So the virus was present, but the genotype was symptomless. So now, then next our aim was that whether the genotype was showing resistance at all the stages of growth, or whether it was susceptible at some particular stages also. So through this study, what we found that our resistant genotype DLS cell 10 and WBC cell 5, they showed a little susceptibility reaction at seedling stage, but as the plant grew, and as it attained the adult stage, the symptoms recovered. The plant, there was a phenomenon of post recovery observed, and full flowering and normal fruit development was there. So basically, what we found that our resistant lines were susceptible at seedling stage, but they show a host recovery mechanism. So based on this, we have a very good publication in Plants, the journal Plants. And next step, what we undertook was to understand the genetics of resistance, for which we had developed F2 mapping population. And here we screened our population both under natural field conditions as well as through challenge inoculation. And through challenge inoculation, we used viriloferous whitefly method for understanding the genetics. And through this, we found that our resistant sources were housing resistant genes which show monogenic recessive form of resistance. So this is what we concluded. And based on in this also we have made some publications in good journals. Then next step was whether we can map the genes or QTLs for resistance to leaf curl disease. Here, because we were at very initial stages, so we used F2 mapping population only for to identify genes, and we had developed linkage map, and based on that, the preliminary study which we have done on the F2 mapping population, we have found that some major QTLs on chromosome number 3, we could locate some chromosome QTLs on chromosome number 3. Apart from that, we found some QTLs which show epistatic interactions, and they also contributed significantly to the phenotypic variants which for resistance to leaf curl disease in the population. So that was a very important result which we observed in this study. And we also did some transcriptomics work where we try to study the expression profile of various transcriptomes upon infection with the varioliferous whiteflies. And we, based on the transcriptomic studies, we try to fish out some candidate genes which may be involved in host resistance mechanism, which included various defense-related genes, some Burki factors, transcription factors, and other genes. So these are some of the real-time expression profiles of the various genes of the transcription factors which we observed. So what we found that the expression of all the defense-related genes was high in the tolerant genotypes post 15 days, because it takes approximately 10 days for the symptom expression. So all the defense genes were activated during the initial 15 days after infection, when we were comparing it with the susceptible genotypes. So we have some publications with respect to this study also. Then the second aspect which we are working is the heat tolerance breeding in chili. So because in North India, as we know that during the summer months, the night temperatures can go more than 25 degrees Celsius, and chili cultivation is severely hampered if the night temperatures are quite high. The optimum temperatures for chili setting is 15 to 20 degrees Celsius only, and any temperature more than 25 degrees, night temperature more than 25 degrees Celsius, severely affects the fruit set. So for this, we had started looking for genotypes, whether any heat tolerant genotypes in chili are there. And in this process, we identified some genotypes which were fixed, advanced, and also evaluated. So at present, initially in this study, we had identified some 17 genotypes which were heat tolerant. Out of this, 2 genotypes, DLS 161 and 152, they were found to be very good, fruiting at more than 37 to 44 degrees Celsius during the day and 25 to 33 degrees Celsius during the night. And we could also register these genotypes with NBPGR New Delhi. And we did some morphological, biochemical, and molecular studies on these heat tolerance genotypes to understand the molecular mechanism of resistance. So what we found that in chili, it is basically not the vegetative stage but the reproductive stage which is affected by the impacted by the high temperature tolerance, high temperature, and which resulted in reduced fruit size, fruit weight, fruit and seed number, and yield of the genotypes. So these reductions significantly lowered the yield in susceptible genotypes. But in heat tolerant genotypes, all these morphological features, the reduction in these morphological features was much less when compared to the susceptible genotypes. So we did some biochemical and molecular profiling where we use some important genes which are involved in abiotic stress resistance. And in this figure, we can see that these genes were significantly overexpressed in the tolerant lines which we had taken, and the susceptible genotypes were showing lower expression. We also studied some heat shock proteins in these genotypes, which were also found to be overexpressing in the heat tolerant genotypes. So we have some publications with respect to these studies also. And then we also have attempted a mapping for heat tolerance in chili, where we have developed the F2 mapping population. And we use the genotyping by sequencing approach to map the QTLs. And we had targeted some 25 traits, which included both morphological, physiological, biochemical traits. And we had developed the linkage map, and some 54,000 SNP markers were mapped during these studies. And in this, out of the after QTL mapping, we identified 63 QTLs, out of which there were some for 14 traits we found QTLs which were having phenotypic variance more than 10%. So these were the QTLs of interest to us. And we also found that because we were studying various traits, so many of these four traits were co-localizing on the same chromosome. So actually it is the same genomic locus which is controlling tolerance to this abiotic stress in chili. So these are some of the studies where we found co-localized QTLs. And we also did the candidate gene analysis where we found that majority of the genes which were fished out after this study, they belong to the metabolic pathways, biosynthesis of secondary metabolites, and basically the transcription factors like the BLHLH family proteins, MIB proteins, ERF factors, NAC proteins. So all these proteins are basically in some way or the other related to the defense host machinery. So we found these genes in our candidate gene analysis also. So in conclusion, we could say that this heat tolerance in chili is quite complex because we identified many QTLs and highly influenced by the environment. And the major QTL which we found was for the fruit yield, which had the highest PV value of more than 19%. So we have this one publication in Frontiers in Plant Science with the work we have done this on mapping of heat tolerance. Then some brief work we have done for cold tolerance also. We did not achieve much success for cold tolerance in chili, but still we have identified some tolerance genotypes where we did some basic biochemical and gene expression analysis, where again, similar to the heat tolerance studies, we tried to identify the major biochemical compounds involved in abiotic stress and also the expression of the genes involved in abiotic stress, where the cold tolerant genotypes showed means overexpression of all the stress related genes. So indicating that the genotypes which we have identified, they have some potential and they can be utilized in further breeding program. So at present, the IRI for the chili breeding program at IRI, we are involved in developing mapping populations like rails for heat, leaf curl tolerance and heat tolerance, nails for heat tolerance, magic populations, and we are also doing GWAS studies for heat tolerance, cold tolerance, and anthracnose resistance. And we have also developed. We are also in the process of developing double haploid protocol in chili, where the haploid production protocol has already been standardized. This is not the work which I have done in alone. It is the entire team which is doing. And the major constraint which I am still facing is the short breeding period at IRI, because of which the large scale seed production for identified promising hybrids, it's still a challenge for me. And for this, I am looking forward for collaborative opportunities with ICR institutes, SAUs, and other organizations for synergistic as well as mutually beneficial partnership, so that we can take the chili breeding program ahead. So the upcoming priorities for chili breeding program at IRI is basically developing heterotic pools in chili, fine mapping of genes for leaf curl resistance and heat tolerance, and developing breeding efforts for export quality in chili at IRI. So with this, I want to thank the director, joint director, and head of the division of IRI New Delhi, and all the lab mates, my colleagues, and the field staff who are working in the chili program. I would like to thank them. And at last, again, I would like to express my sincere gratitude to the Society of Plant Research for conferring me this prestigious fellow award. Thank you so much, sir.
Thank you, Dr. Arpita, for a wonderful presentation. And the work you have already done and the work which we are pursuing further. Getting an award is an encouragement to the young scientists, especially, so that they should do still better in the time to come, because this is just a beginning. We have to achieve many more things, and let me congratulate you once again. Thank you very much. And I would request Dr. Naseem Mohammed to kindly be here with us, because Dr. Akhilesh Sharma has received a request from Dr. Akhilesh Sharma, because he is having some confidential work to move to Delhi. So can we simply shift your lecture immediately after Dr. Sharma?
Okay, sir. No issues.
Thank you very much. So I would request Dr. Akhilesh Sharma to kindly give his presentation. He is professor in Department of Vegetable Science and Floriculture, College of Agriculture, CSK, HPKV, Palampur, Himachal Pradesh. And one thing I would like to mention that because we are running short of time and we are having paucity of time, because many more lectures are to follow. So kindly restrict your lecture within the time provided. So it will help others. Dr. Akhilesh Sharma, please.
Thank you, Dr. Subodh Bhatnagar for kind of allowing me to present my work before the scheduled lecture. And I thank Nasir sir also for giving me this his slot for presenting my work. So thank you very much. First of all, I thank Society for Plant Research for conferring me this prestigious fellow award. This is really a great opportunity for me, and acknowledging my contributions, I definitely convey my thanks to all kind of authorities of Society of Plant Research.
Please.
So should I start, sir?
Yeah, please.
Thank you very much, Dr. Subodh Bhatnagar, sir. My topic is breeding pea for powdery mildew disease resistance and male sterility based hybrids in chili and cauliflower. I'm presenting on these three crops because I am working on these crops. So I just want to share my research work and the kind of contributions in this area of pea, chili, and cauliflower. Next slide, please. So as far as powdery mildew resistance in garden pea, as we know, this is one of the serious problem. In garden pea, there are many diseases, but this is one of the serious disease. And in pea, there are certain demands from the consumers as well as from the different private sector companies. So major requirement of farmers to have long pods with proper filling and dark green colors. Beside that, sweetness and shelling percentage, that is also very important. So vis-a-vis powdery mildew resistance, I am simultaneously working on these aspects also. Next slide, please. So as far as powdery mildew resistance is concerned, there are 3 genes which have been reported. 2 are resistance and 1 is dominant in inheritance pattern. And this particular, this dominant ER3 that has been successfully introduced from Pisum fulvum. But as far as my work is concerned, I have no source of ER3, but I have ER1 and ER2 genes which I have transferred in my material. Next slide, please. When I started working on pea crop, I used both conventional and non-conventional breeding approaches to improve certain traits and to develop high-yielding varieties. In non-conventional breeding approaches, I also worked on mutation breeding, as I got one of the projects from ICR and simultaneously from Bark Mumbai also. So these non-conventional techniques I have used in my work. Next slide, please. As far as conventional breeding approaches, I followed hybridization and followed by pedigree method of selection. And through this, I have kind of developed 2 varieties. One is Palm Triloki and Palm Smooth. So the major focus was to develop varieties with the long pods, well filled, that is more than 10 seeds per pod, and sweetness, as I have already discussed with you. And this ultimately resulted in the development of these varieties, but there are certain lacunas in the Palm Sumul. To improve that variety, we crossed that with Palm Priya, one of the important variety, and we able to develop a new variety that is Him Palm Matter 1. And this has been kind of recommended for Zone 1 of the country also, that comprise of J&K, Himachal Pradesh and Uttarakhand. And simultaneously, another line that we have isolated from our crossing material, that is heme polymeter 3. This has recently been recommended for cultivation in the state, and it is now put forward. Its recommendation proposal is put forward the State Varietal Disease Committee. So for this screening of powdery mildew resistance, we have very hot spot in Himachal Pradesh, that is in the zone 4 in district Lahaul and Spiti. So we screened our material there at Kukumseri and simultaneously under lab conditions also, so that to be able to develop good resistant varieties which have a better adaptability, and they show resistance over the environmental conditions. So following these techniques, we have able to isolate this DPP SP6. This is now released as heme polymeter 3, and this DPP SN1. This we have recently put in All India Coordinated Research Project for evaluation, and hopefully we will get some good results from there also. Next slide, please. Next slide. So see this heme polymeter 1, as I have told you, this is one of the very good variety, and now in our Himachal Pradesh, almost whole area under P, that is, it is a #1 vegetable crop in Himachal Pradesh, being grown throughout the year in one or the other region and cultivated as an off-season crop. Now majority of the area that is under this variety, and seeing this popularity and good characteristics, 16 private companies they have signed MOU with our university for multiplication of its seed and further sale to the farming community. And see the size and feeling of this variety. And on the right side of this heme polymeter 3, and farmers, they are also demanding now seed of this variety also, because it contains maximum 13 seeds and on average 10 to 11 seeds, and pod is quite larger, bright in green, and quite sweet. Next slide. Next slide, please. Next slide, please. Previous slide, please. So as I have told you that I am working on these molecular approaches also. So this under this slide, through mutation breeding, we have isolated 3 lines, that L401014, L0.3139, and AP0.3129. These 3 varieties they have been isolated through mutation breeding, and this L is for Lincoln and AP is for Azad P1. So these are these 2 variety, Lincoln and Azad P1, they were mutated through physical mutations as well as chemical mutations, and through that we able to isolate these 3 lines. And these now this paper is published recently in scientific report, printed in Plant Sciences, and that is one of the good journal. And next slide, please. And these lines, they are validating ER1 and ER2 markers simultaneously. So we are working further to establish whether these lines, they carry ER1 and ER2 genes. But presently they are validating the markers which are associated with these ER1 and ER2 gene. Next slide, please. Now, with this, I have this is the work I am doing in P. Now I am also simultaneously working on cauliflower, and cauliflower, now farmers, they are kind of demanding hybrids, since majority of the hybrids which are being cultivated in the farmers field, that is through private sector companies, and seed cost is quite high, and our kind of government want that universities should come forward to have hybrids in these crops also, as it is also one of the off-season crop being grown throughout the year in our state, and quality is quite good due to the favorable climatic condition in the state. For initiating work for this hybrid seed production, I tried to transfer this CMS system in our own varieties, and we isolated 4 CMS lines in cauliflower mid and mid, late, and late group of this cauliflower, because we have the kind of monopoly or dominance in late cauliflower, as it needs temperate conditions, low temperature condition for seed production. So keeping that in view, we worked on this this system, CMS system, and we isolated 4 lines. Then we utilized these lines in hybrid seed production. Next slide, please. And be able to isolate 2 hybrids, which are recently being recommended for cultivation in our state. That is Himpalam Fulgobi Hybrid 1 and Himpalam Fulgobi Hybrid 2. Both they are belong to mid-late group. And Himpalam Fulgobi 2 is around 10 days earlier than Himpalam Fulgobi Hybrid 1 in curd formation. So if we grow these 2 hybrids simultaneously, then they able to prolong the duration of the crop, and simultaneously that the glut can be avoided and farmers can get good price in the market. So this is the reason that we have simultaneously released these 2 hybrids in cauliflower. And these hybrids being compared with one of the very popular hybrid from the private sector company Maharani, and these both hybrids, they're showing superior performance to these private sector hybrid, that is Maharani, and farmers, they are demanding the seed of these hybrids. Next slide, please. Because time is short, so I have just tried to give the glimpse that the kind of the good results I have obtained in these crops. Simultaneously, in chili also, there are no hybrids which are released in the state, and we have very serious problem of bacterial wilt and simultaneously fruit rot. Since in certain pockets of the state there are heavy rains, and the problem of fruit rot is quite high. So there is unique characteristic, unique varieties are required which has erect fruiting habit. That those varieties or hybrids, they have low incidence of fruit rot. Accordingly, we planned our research program keeping these aspects in view. Next slide. So we develop 4 GMS lines in chili in our own material. We transferred this male sterility gene, and now I'm simultaneously working on transferring this CMS system also, and lines, they are with me, and we are now utilizing those lines also in our hybrid breeding program. But through this GMS system, we have released 3 hybrids in the state, that is Himpalam Chili Hybrid 1, 2, and 3, and Chili Hybrid 1 that has erect fruiting habit, whereas 2 and 3, they are drooping habit. Particularly in the zone 1, that is the low hills of the state, because they have a little high temperature in June to August, and there is problem in erect fruiting habit, that there is a scorching heat that you can say damage the color of the fruits, and they want this drooping habit because foliage that protect the fruits from scorching sun, heat and light. So these are the hybrids. They have been accordingly released in the state using male sterility system. Next slide. So with this, thank you very much. This is the kind of work I have done in these 3 crops. Thank you very much.
Thank you very much, Professor Sharma, for a wonderful presentation. And this cauliflower is an important vegetable, and particularly in the area where you are working. The biggest threat in Himachal is the use of pesticides, which everybody is fighting against, and biocontrol measures are being adopted. So we wish that the things are improving at Himachal now, and the use of pesticide has been reduced to a greater extent. So thank you very much for a wonderful presentation.
Thank you very much. Thank you for giving me the opportunity.
I wish to congratulate you once again for being conferred to this fellow award. Thank you very much.
For showing me this prestigious fellow award.
Thank you. Thank you,
Thank
Professor
you.
Sharma. Now I would request Dr. Nasir Mohammed to kindly continue with his presentation. And at the outset, let me congratulate him for being nominated for this prestigious award. Dr. Nasir.
Sir, good afternoon, sir.
Yeah,
Sir,
Dr. Nasir Mohammed is scientist E and head, Genetics and Tree Improvement Division, ICFRE, Tropical Forest Research Institute at Jabalpur. Professor Nasir, please.
First of all, I am thankful to chairman and esteemed jury members for considering my nomination. Very good afternoon to all of you. I am Nasir. I am forest geneticist working at Tropical Forest Research Institute, and I am working on conservation of forest genetic resources. And for this lecture, I have chosen one species, that is Pterocarpus marsupium. We are working on almost 65 species under the National Program for Conservation of Forest Genetic Resources of Central India. And I'm presenting one case study on Pterocarpus marsupium. Next, please. Before jumping to the research work, I will take this opportunity to briefly introduce my institute. We are located on the banks of River Narmada in Jabalpur. We are a specialized institute for forestry research. Our jurisdictional area is Madhya Pradesh, Maharashtra, and Chhattisgarh. We are addressing the research-related issues of forest of these 3 states, and we are covering the 5 forest types. Our major area of works are FGR conservation, tree improvement, rehabilitation of degraded forest land. Next, please. As you all know, there are specialized institutional mechanisms for plant genetic resources, like in agriculture, but FGR remained neglected till recently, and now government has initiated large programs for conservation of FGR. FGR is very essential for this tract because Madhya Pradesh, Chhattisgarh, Maharashtra, Jharkhand, tribal population and peoples residing in forest ranges are dependent on the FGR for their livelihood. Also, FGR has a great potential to adapt to the climate change. They are the sources for biotic and abiotic stresses, and it will. That's why now the policymakers are giving due importance to the FGR. Next, please. Here I have taken one species to present, that is Pterocarpus marsupium. We call it as a Bija or Vijayasar also. It is a native to our country, and it is its timber is ranked next to the teak and latifolia, rosewood. Although its timber is also important, it is very much important for medicinal purpose also. Almost all parts are important and used in various medicines, but its hardwood is in very great demand because it is used in managing the diabetes. Its hardwood contains the marsupiin and pterostiglin, which has a equal potential or greater than the metmorphin, and that's why it is extracted very unsustainably. And now this species is included as a near threatened in red data book. So we got the project. Next, please. So we have undertaken this study to check what is the status of natural regeneration. Whenever we used to propose the conservation strategy for forestry species, we have a standard procedure to check what is the status of natural regeneration by laying out the sample plots, how much seedling, sapling, adult trees are there in the natural forest, which are the factors affecting their, and how to manage that factor so that regeneration is profuse. What is the variability for morphological and biochemical traits, whether the genetic clusters are there or not, and to develop the propagation protocol. These are our standard procedure whenever we used to take the work on forestry species. And then we used to go for the conservation strategy. For sampling or selection of study sites, we have consulted the working plans. Working plans are the documents which lies with the forest department. These are the government documents, and where the species distributions are were very well documented. So we consulted these documents, and we have followed adaptive cluster sampling to select the study sites in deciduous forest of Madhya Pradesh. Next, please. So for natural regeneration, actually natural regeneration is nothing but the process of sylvigenesis, where we can check how the species is surviving over the years. So for this study, we have laid out the selected 7 study sites, laid out 46 plots, each measuring 0.1 hectare in this forest area, deep inside the forest area. These study sites belongs to the almost different forest division: Mandla, Balaghat, Rewa, Dindori, Katni, where this species exists. Next, please. And we assess the natural regeneration. There are standard procedure to assess the natural regeneration. Whenever seedling is greater than the sapling and sapling is greater than the adults, we said the species is surviving very well. When there is a disturbance in this equation, we need to think, we need to take the conservation measures, and we used to recommend accordingly to the State Forest Department to initiate the actions. So we have assessed the natural regeneration. We have also checked which are the factors those are affecting the natural regeneration through cumulative disturbance index and its correlation with the natural regeneration. Next, please. These are the we have conducted these monitored the seedling, sapling, adults for 4 years, and what we have found, the situation was very we need to take immediate action, because sapling stage was almost absent for this valuable species, native indigenous species. Seedling was there, but seedling was failing to convert into the sapling. Adult was there. We have found 2 study sites, that is Bahuriband and Sara, where seedling was increasing over the years, but these seedlings were failing to convert into the saplings. So, next, please. This graph presents the clear picture how seedling density was varying from year to year at each study sites. Here we can see that Sara and Bahuriband, the seedlings were increasing. So we have requested the Forest Department to undertake the assisted natural regeneration work, so that the seedling can be converted into the saplings. Next, please. We have to check the parameters which are disturbing the natural regeneration. We have recorded the number of villages in 10-kilometer radius of the study sites, the population of that villages, livestock of that villages, weed infestation at that site, and we have categorized our study site as a disturbed, undisturbed, and highly disturbed. Next, please. Then we have plotted these parameters against the seedling density, and what we have found that the weed infestation is affecting the seedling density, followed by the lopping and cutting of the branches by the people residing near the forest, followed by the livestock and human populations. And as you all know now, forest area is now getting infested with lantana, hyptis, one tulsi. So the native species are regeneration is greatly now affecting. Next, please. These are the glimpses. As we belongs to the Forestry Institute, we have access to the deep inside forest. So we have laid out the permanent plots for 4 years. Next, please. We received the support of State Forest Department. Next, please. Next, please. Next. To assess the variability, we have also recorded the data on various morphometric traits. Then we have also done some molecular studies. Next, please. As we know, for forestry species, genomic resources are not available easily, so we use the available markers. Next, please. What we have found that in this species, there are a very high range of variation for almost all the traits. So there is a scope for selection of good genotypes. So we have also practiced and selected promising genotypes, multiplied them, and established the germplasm banks. Next, please. We have done the location-wise analysis also. Even in location-wise analysis also, we got the high range of variability. So we have recommended some study sites like Sara, Bahuriband for the conservation purpose to the State Forest Department. Next, please. These are the glimpses for working in the forest areas. Next, please. Molecular study revealed that although the clustering we have seen, but there was no clear-cut clustering. So we have concluded that the population of marsupium was continuous, and recently it got fragmented due to the over extraction. Next, please. Even one of my member also estimated the pterostablin range of pterostablin, so that the populations with high biochemical active compounds can be identified and suggested for conservation. Next, please. GIS team of our part checked how this species is going to perform under climate change scenarios, under different conditions, and they have concluded that. Next, please. That is major potential promising areas lies in the central India only, followed by southern and western India. In 2050, the modeling suggested that this species can move towards the northward shifting. Its population is going to reduce in the eastern zone. Also, the annual precipitation was the only factor which are going to affect it. It is a limiting factor for this species distribution. Next, please. So we have also developed its propagation protocol. Its seed coat is hard, so we found the physical scarification or soaking of seeds are greatly enhancing its germination. So we have published this one also. Next, please. We have undertaken the multiplication of selected trees. We have established their germplasm banks at Jabalpur, Chhindwara, Raipur, as this species is very much important, and MP and CG categorize this as a promising species for conservation aspect. Next, please. So what we have suggested to the State Forest Department that Sara, Simaria, Lamta and Bahori, our study site, should be considered for the conservation as it is a non-domesticated species. In situ conservation is the most viable approach. Genetic study revealed that recent fragmentation in its natural habitat is due to the unchecked biotic pressure. We have suggested Forest Department to take the assisted natural regeneration because seedling was failing to convert into the sapling. So this methodology will be very much helpful for them. Next, please. Next, please. And we also suggested them that the Forest Department used to run the big programs like Haryali Prasar Yojana, National Afforestation Program. So include this species in their program because presently they are planting the teak, eucalyptus. So this native indigenous species, we have recommended them to include in their programs. And we have recommended tall saplings should be preferred for the plantation based on our study. Next, please. Other than this species, as I earlier told, almost 65 species which consist of timber as well as non-timber species, gum-yielding species. These are the wild species on which we are working for its conservation, propagation, and improvement aspect. Next, please. We have strong development tree improvement program, and we are targeting this species: Tectona, Latifolia, Buchnania, Madhuka. As we all know now, these non-timber species like Chironji, Mahua, they are declining rapidly in the natural habitats. Next, please. We have germplasm banks trials across the 3 states. Next, please. Like in agriculture, they used to call superior genotypes. Here we used to call the candidate plus tree. So we have the candidate plus tree marked for almost all the important species. Next, please. We have a strong team working on the macro propagation aspect. Forestry trees are difficult to propagate clonally, but we have almost developed the propagation protocol for the important species. Next, please. This is the team at my division. We are working. Next, please. These are the publication on Vijayasar, that is Pterocarpus marsupium from this study. Thank you. We are open for the collaboration with the teams or researchers who are interested to work on the forestry tree species, wild forestry tree species, including gum yielding, wild fruits, etc.
Thank you very much, Dr. Nasir, for a wonderful presentation. And forestry is one very important area where you will hardly find more experts.
Yes,
so
sure.
certainly Vegetos will take advantage of it and will certainly seek your expertise as and when required. Thank you very much.
Thank you. Thank you.
Thank you and congratulations once again.
Thank you, sir, for considering.
Now I would request Dr. Yogendra Kalanahaldi. He is scientist, cell and molecular biology and Trait Engineering at ICRISAT, Hyderabad, Telangana. Dr. Yogendra, please.
Yeah. Thank you, sir. Can you see my screen?
Yeah, it is visible very much.
Yeah, I'm Yogendra Kalinalli. I'm working as a scientist at ICRISAT. Today, I'm going to talk on leveraging multi-omics to predict and mitigate plant pathogen interactions. So it is estimated that by 2050, world population will increase to 10 billion people. To feed this increased population, we need large-scale step change in the crop production. However, crop productions are continuously affected by biotic and abiotic stresses, and the recent climate change has an adverse effect on the global yield of major crops. So how we can improve the crop yield? So recently there's a 5G concept has been proposed to improve the crop genetic improvement. These 5Gs are, first G is the genome, availability of the genome sequences. Now most of our ICRISAT crops having the complete genome assembly available for all the ICRISAT crops. And second G is a germplasm, availability of the germplasm that include the wild relative land races which have been characterized at agronomical level. And third G is the gene, identification of the gene and functional characterization of those genes. The fourth one is genomic breeding, using the gene information, like a genome information, how we can utilize that genome information for crop breeding improvement, especially mainly for the genomic knowledge, genome by sequencing or a GWAS data. And fifth G is the gene editing to improve the plant performance. So in my talk today, I'll focus mainly on aflatoxin contamination in peanut. Aflatoxins are the secondary fungal metabolite produced by the fungus Aspergillus flavus and Aspergillus parasiticus. There are more than 20 aflatoxins are reported. Among them, AFB1, B2 produced by Aspergillus flavus, AFB G1 and G2 produced by Aspergillus parasiticus. These are the major ones. Aflatoxin toxicity, it ranges from acute effects. Sometimes it causes death to the chronic effects like liver cancer. The regulations of aflatoxin contamination in food varies from country to country. In India, it's a permissible level is less than 15 ppb. However, in Europe, it's less than 4 ppb, but in US, it's less than 20 ppb. Apart from groundnut or peanut, cotton, sunflower, wheat, corn, rice are the other key hosts for this Aspergillus flavus causing the aflatoxin outbreak. So far, there is no commercial variety available for aflatoxin resistant. So what we have to do is we have to utilize the advances in biotechnological tool to combat this aflatoxin problem. In our lab at ICRISAT, what we did is we used a double defense approach to prevent the aflatoxin mitigation. First approach we used is a overexpression of the defensin gene. Defensin is an antimicrobial peptide. When we overexpress defensin gene, it creates a pores on the fungal cell wall. That way, it prevents the fungal growth. Second approach we used is host-induced gene silencing approach. Here, the 4 fungal specific genes were overexpressed in groundnut. When the pathogen infect, these fungal genes activated, they silence the fungus gene. That way, aflatoxin production is reduced. However, the aflatoxin resistant is a complex and quantitative trait. Moreover, the transgenic approach is not social acceptance, and also a lot of regulations is required. So to overcome this, what we used is we use the multi-omic approaches to understand the mechanism. So far, there is no molecular and biochemical mechanism is understood in groundnut and Aspergillus flavus interaction. What we did is we have the phenotype. Phenotype is the transgenic lines. Transgenic lines is giving a low aflatoxin contamination. To use these transgenic lines, we used a forward genetic approach or a bottom-up approach using metabolite, then integrated with the proteomics data. Then we identified what are the genes controlling this resistant or what genes are causing more of susceptibility. So in detail, if you go to the transgenic groundnut, expressing the RNAi genes. Here we, for the Higgs lines, we used 4 RNAi genes, like 4 fungal specific genes. These are NSDC, VIA, AFLR, and AFLM. These are from the aflatoxin pathway gene. Here you can see AFLR is a transcriptional regulator gene. NSDC and VIA, they are involved for the aflatoxin developmental genes. They mainly involved for conidial germination, sclerotia production. And we selected one more gene, AFLM, that's from the aflatoxin biosynthetic pathway. When we silenced all these 4 fungal specific genes, we developed the transgenic lines, RNAi lines. Here you can see these 2 are the transgenic lines expressing the 4 fungal specific genes. Here the fungus growth is less compared to the null mutant or a wild type. And also we measure the fungal load. Fungal load is less in the transgenic compared to the wild type, and also aflatoxin contamination, which is less than 15 ppb in the transgenic lines. So we use these lines for the metabolomic work to understand what kind of biochemical mechanism involved. Here we use the resistant is a 4 RNA transgenic lines, susceptible is a wild type ICGV 91114. We did experiment in 2 time point, 48 hours and 72 hours post-infection, 2 treatment, pathogen and mark. Mark is a water inoculated sample. We got total 4 samples, resistant path, resistant pathogen, resistant mark, susceptible pathogen, susceptible mark. We did the experiment with 3 biological replication. Then these samples were subjected for non-targeted metabolomics analysis using liquid chromatography, high resolution mass spec. When we got the data, first what we did is we did a multivariant analysis or a principal component analysis to know how these groups are, whether these all the 4 groups, resistant pathogen, mark, susceptible pathogen, and mark, whether they form the group or to know these things, we did a principal component analysis. Here you can see PC1 that separate 40. It explains 48% of the genotypic phenotypic variation. It can clearly separate the genotype. These 2 are the susceptible sample. These 2 are the resistant sample. PC2 it separate 22%. Here you can see it separates based on the treatment. These 2 are the pathogen treated sample. These 2 are the mock treated sample. So we identify the metabolite. There are 2 kinds of metabolites are identified. Resistant related constitutive metabolite. They are called as a RRC metabolite. These RRC metabolites obtained by comparing the resistant mark with the susceptible mark sample. Second group we identified is resistant related induced metabolite. They are called as RRI metabolite. For this, first we compared the resistant group RP by RM. That whole group compared with the susceptible sample SP by SM. From this, we identify the metabolite mainly from the phenylpropanate pathway, flavonoid pathway, alkaloid, and fatty acid pathway. We can't focus for all the pathway. What we did is we identify a high fold change significant metabolite, mainly from the phenylpropanate and flavonoid. Some examples are ferrulylagmatin, we identified as a 9.4 fold change as a RRI. In FEL putrescine, coniferal alcohol, coniferal aldehyde, synapyl alcohol. Same way for flavonoid, we identified petunidine, naringenin, epicatechin. If we see the detailed pathway for the phenylpropanate pathway starts with L-phenylalanine. Here it produces from the shikimate thing, shikimate in the from the TCA cycle. So it produces shikimate converted to phenylalanine. Phenylalanine further converted into cinnamic acid converted into coumaric acid, caffeic acid, ferulic acid, hydroxyferulic acid, and synapic acid. These coumaric acid to synapic acid, these are called as a hydroxycinnamic acid. From 1970-1980 onwards, these metabolites are reported as either antioxidant or antifungal or antibacterial property. But in our study, what we observed is this hydroxycinnamic acid further converted into hydroxycinnamic-CoA thioester, like coumaric acid converted into coumaryl-CoA, caffeic acid, caffeil-CoA, ferulic acid, ferulyl-CoA, same way synapic acid, synapyl-CoA. These CoA thioester further converted into aldehyde and alcohol, like coumaryl-CoA converted into coumaryl aldehyde, then coumaryl alcohol, that form the lignin, H-lignin. Same way, ferulyl-CoA and synapryl-CoA further converted into their respective aldehyde and alcohol compound metabolite. Then they they form the G and S lignin. From various previous plant pathogen interactions or in various biotic stress, we observed lignin is the polymer compound. It deposits on the cell wall. It helps for the secondary cell wall thickening. Parallelly, this hydroxynamic acid CoA thioester, they combine with amines. Amines come from the amino acid from the TCA cycle. Here you can see shikimate further converted into tyrosine. In the TCA cycle, we can observe the arginine. This ornithine, arginine, tyrosine, these amino acids converted into amines. Putrescine, arginine converted into agmatine, tyrosine to tyramine. These amines combine with HCA-CoA thioester, they form the hydroxycinnamic acid amides. These are the conjugated compound, conjugation of CoA thioester and amines. For example, agmatine combined with coumaryl-CoA, it forms the coumaryl-agmatine. Same way, coumaryl-tyramine. So this conjugated compound in various plant pathogen interaction, it's reported this hydroxynamic acid amide deposit on the cell wall, help for the secondary cell wall thickening. To prove this hydroxynamic acid amides and lignin deposition, we did a histochemical analysis. For lignin, we used a fluoroglucinal HCl stain. Here we used a 4 sample: resistant pathogen, resistant mark, susceptible pathogen, and susceptible mark. Here in case of resistant pathogen, here you can clearly see the pinkish, dark pinkish fluorescence, which is more predominant in RP sample compared to RM and SP and SM sample. This shows that in case of resistant pathogen infected sample, lignin is depositing more, that help for the cell wall thickening. Same way, for hydroxynamic acid amides, we used a new reagent for the histochemical assay. Here again, we used the 4 sample RP, RM, SP, SM. Here in case of RP, you can see the blue fluorescence, which is more predominant compared to RM, SP, and SM. This shows that lignin and HCA, they deposited more in case of the resistant pathogen sample, that help for the secondary cell wall thickening or cell wall reinforcement to prevent the pathogen entry. After the metabolomic analysis, we use the same sample for the comparative proteomic to know what kind of the resistant related proteins or a susceptibility related proteins involved in the Aspergillus flavor inspection and also for the aflatoxin production. From this, what we did is like we identified the hierarchy of the genes, receptor like protein kinase, argine, NPS-LL argine, TIR, NPS-LL argine, phytohormone, ethylene, axine, ABA, transcription factor, MIP, MAC, ethylene responsive factor, MATS, and from the secondary pathway, mainly phenylpropanide pathway and also fatty acid pathway we observed. So when we integrated both metabolomics and proteomics, help us to identify the hierarchy of the genes. Here the Sankey diagram clearly shows how these different groups like proteins and the metabolite group from 48 and 72 hours, both RRA and RRC, how they separate into the pathway and how they like integrated as a differentially accumulated metabolite or a differentially expressed protein. From this integration, what we did is like we can clearly explain what is qualitative resistant and what is quantitative resistant involved in the groundnut Aspergillus flavus interaction. Here, this if you see this figure, when the groundnut peanut is contaminated this with Aspergillus flavus, the pathogen Aspergillus flavus, it produces certain pathogen associated molecular pattern. They may be either some proteins or cell wall degrading enzyme or some metabolite. To prevent this, plant produces a receptor compound. They are called as a PRR compound, like a pattern recognition receptors. That resistant is called as a PTI or PAM triggered immunity. This is the first level of defense. Most of the time, pathogen can easily overcome this PTI by production of the A virulence factor and virulence factor. To prevent that, plant produces the NBSLR genes, NBSLR genes. This resistant is called as the hypersensitive resistant or effector triggered immunity, or called as a qualitative resistant. It's a monogenic resistant. Some of the specialized pathogen like Aspergillus flavus that can easily overcome this ETI by production of more of a virulent strain. that time plant activate the durable resistant or a quantitative resistant by activation of the MAP kinase. Further, it produces the phytohormone transcription factor. Then it activates the fatty acid, like a secondary metabolic pathway, fatty acid, flavonoid, and phenylpropanide. Finally, it will help for the secondary cell wall thickening. So from this interaction, what we did is like we identified the susceptibility associated gene, which are responsible for more of Aspergillus flavus infection and causing more of aflatoxin contamination. Now what we are doing is like we are using the susceptibility genes as a potential target for CRISPR-Cas9 based gene editing. So before starting silencing the susceptibility associated gene, first we standardize the CRISPR-Cas9 based gene editing in groundnut by silencing the phyton desaturase. Here you can see the edited and non-edited plant. Edited plant is a clear albino. This PDS is from the phenylpropanide pathway. When we silence PDS, it creates a albino phenotypes. And further, this one of the gene we are using is an annexin gene. We designed the guide RNA from the exon 4 and 5. Here you can see we have got different kinds of the edits and larger deletion as well. Finally, we observed the 24% knockout score. We are currently doing the phenotyping for this aflatoxin resistant for the edited plants. With this, I would like to thank our team ICRISAT and collaborator from Australia Department of Primary Industries and also Western Sydney University. Finally, I would like to thank Society of Plant Research for considering me for this prestigious Fellow Award 2025. Thank you.
Thank you very much, Dr. Yogendra, for a wonderful presentation. And let me congratulate you once again for the
Thank you, sir.
lecture and for the award, which is well deserved. And I'm sure that Vegetos will be in collaboration with ICRISAT for certain publications and certain collaborative programs. Thank you very
Definitely,
much.
sir.
Dr.
Yeah,
Thank you very much.
definitely, sir. Thank you, sir.
Thank you. I would now invite Dr. Chandan Singh Chanotia. He is Senior Principal Scientist, Phytochemistry Division, and Professor in the Faculty of Chemical Sciences, CSIR CMAP, Lucknow. Dr. Chanotia, please.
Hello, sir. Am I audible, sir?
Yeah, you are audible, Dr. Chanotia. Yeah,
Thank you so much, sir.
welcome.
Thank you so much, sir, for inviting me in today's, the second day, particularly for this Fellow Research Award.
And our sincere greetings to you. And
Thank you, sir. Thank you so much. So I will not take much of your time, and I will simply talk on. The subject is very well known for most of us as far as as far as chemistry of these aromatic plants are concerned, and most of the publication basically that depends on these secondary metabolites. So I will talk about the structural diversity in essential oils and how we can authenticate those essential oil based on the available tools. So, beta जरा इसको कर दो, इसको जरा full screen कर दो. Yes. So now, before talking detail about the diversity in essential oils, I want to have just bring to your notice the famous quote given by the Nobel laureates in chemistry. He says that when I draw a molecule in China or in Argentina, it is the same molecule. Okay, people understand immediately without knowing the Spanish or the Chinese. That is this is the beauty of what you call the chemistry. And the only common goal is not about power or borders of the country. It is about bringing forward human knowledge. So Ben has talked about this when he got the Nobel Prize. So inspiring of his message, what I outlined my talk on structural diversity in commercial important essential oils. So basically, I have chosen 3 oils: vetiver, patchouli, and ocimum. And I will give you a brief outline what kind of structural diversities are present in those plants, and how to deal with the authenticity assessments. So if you see, if you take a brief background of essential oils and their current status, so basically these are low molecular weight lipophilic constituents which have a property to volatilize at relatively low temperature. They are actually comprised of a lot of compounds, so that's why we call them complex mixture, and mostly they are composed of mono, sesqui, terpenes, phenylpropanoids, and acetylenic compounds. So a wide variety of structures are present in any essential oil. So what is the advantage? Why there is a need to work on these oils? Because of their immense commercial value as flavor enhancer or as fragrance ingredients. You see a lot of fragrance based cosmeceutical products, consumer products. The essential oil is one of the ingredient you can see in those products. So as already I have talked about why these, what are the essential oils? They are low molecular weight essential components, and they have very immense commercial value as flavor enhancer or as a fragrance ingredients. So the market is flourishing very rapidly post COVID, or you can say due to the post pandemic, because of several because of its association with health benefits. So extensive use, say for example, the citrus oil is one of the example which has been extensively used in a lot of home care products due to its antibacterial nature. Now where is the problem lies? We have enough market, lot of growth is there, but the only problem lies because we have limited we need the demand is very high, production volume is also need to be very high. They are not reaching the consumers in their original composition. So there is why not reaching? Because of the demand. A lot of demand is there. There are a lot of products, consumer based products, which need these essential oils as ingredients. So what actually the middleman is doing, what the industry is doing, they are adjusting the these oils to meet the required quality or the specification. Specification, if I say, I will talk about the international specification that is governed by ISO, International Organization for Standardization, or the Indian regulatory body, that is Bureau of Indian Standards. So these bodies are, although they have a regular watch over the specifications of these oils, but still there is a lag where these adulterants or the adulterants are being added gradually just to meet the demand. So there is a question, because we talk that these are one of the important ingredient of perfumery or they are being used extensively for fragrance creations. Why some of the oils are why they are so esteemed basic ingredients? Because they have very complex fingerprint, they have unique and long lasting odor profile, and one of the very important parameter that is they are impossible to duplicate by synthetic route. This is one of the feature where some of the high value oils, they are actually surviving till date as far as the market is concerned. So these are some examples like vetiver, patchouli, agarwood, and sandalwood. These are you can say they are very highly esteemed basic ingredients. So see, if I talk about vetiver, it is considered a basic ingredient of perfumers palettes, but the constituents contributing its odor are not completely known. Though there is a standards, if you talk about international standard, we have an ISO standards which deals with the quality of vetiver for markers like Cushimol, vetivinins, like that. And the major producers are Haiti, China, India, Brazil, and Indonesia. Other important essential oil bearing plant that is called patchouli or Pogostemon cablin. It has also high demand in perfumery, mostly imported from Indonesia. And there is of course a regulatory body, regulatory standards, which facilitates the authentication of or the quality of the oil. So if I want to summarize the vetiver journey so far done in CIMAP in Lucknow, because as far as my institute mandate is concerned, we are dedicatedly work on medicinal and aromatic plants for varietal development, for product development, or just to have quality control fingerprints out of that. So as far as the journey of vetiver in CIMAP concerned, because this is one of the most valuable plant material, the global market is valued around $88 million USD by 2025. And if you see, this is the oil which is present in about 36% of commercial perfumes. If you are a habitual user of using those commercial perfumes, so please recall it that more than 36% of commercial perfume, they are using vetiver as one of the ingredients, just to get the natural olfactory note for the benefit of the consumers. And largest consumers are US and Europe. India, if you see the market, around 16K is the price for Indian, a little less for Haitian, and of course, the least price is being fetched by the Java type vetivers. So what are the limitations? As I told, supply issue is one of the primary criteria, and other sources of high quality oil are limited. Indian vetiver has vast potential as far as its chemistry is concerned. So far, it has been explored very less, and we need to have to work on this plant. And as far as standards are concerned, there is no standard available as far as ISO is concerned. Of course, we have one Indian standard on that. So if you see, this is how the odor of the vetiver oil lies. It has woody note. If you see the woody note, of course, it again breaks into ambery, vetiver, and sandal kind of notes. So if you see, if I want to summarize the things, one plant has everything, a number of sub notes, or you can say undertones are there. So these perfumery industry, these pharmaceutical industries are utilizing these natural materials just to have consumer appear, what you can say, just to have just to develop a compound, or just to develop a product which have consumer acceptance. If you see what we have done as far as the chemistry or the variability is concerned, we have isolated number of molecules starting from Turing systems that we call bicyclic. You can see on the left top of your screen, and of course to tricyclics. We have a number of compounds bearing three ring systems. So this much complex is the vetiver of the Indian vetiver, I can say. That's why it still has a favorite, what you call a favorite brand for perfumer while making the perfume-based products. If you see the other part, apart from the fragrance value, if you go to the biological actions, these vetiver-based fractions are very highly active, and they have shown vasorelaxation activities, as I have shown in as one of the example where I have isolated this molecule and subjected for vasorelaxation studies, and I found it's giving very good dose-dependent vasorelaxation in rat. Apart from that, if I go to the second major plant which bears patchouli, famous patchouli oil, it is also having very complex structures, starting from one ring systems, you can say on the top, which we call bisabolane type of structures. We got some sesquiosabenine kind of structures, and apart from that, we have a variety of guanine type of structures having one 7-member carbon containing molecule, and other having 5 carbon containing molecules. So this sort of diversity is there, it exists there, and of course these Indian oils have good biological action. Like this particular oil inhibits the growth of Malaysia for it's one of the famous hair fall causing organisms, and these oils are very good in terms of their anti-inflammatory potential. They significantly reduce the TNF alpha in treated cells. We have done these studies and published the work in many journals. Now coming to one of the most precious plant, that is called basil. Indian basil is very popular in one or other ways. We studied a lot of basil species, and our breeding department people, so our scientists in breeding departments, they have developed hybrid osimums. So by making them hybrids, now we are able to cultivate these hybrids for many years. They are because in the usual you see osimum is an annual crop. Its habit is annual, but nowadays by extensive research we have done and our fellow colleagues has done over here, and we have the lines, we have the varieties which are cultivable for perennial habits. So we have, as already Dr. Yogendra talked about phenyl proponites. So because our scientists have developed a lot of interspecific lines by hybrids, and successfully we have characterized one molecule, that is positional isomer, and what we call as a meta-usinol. This was the molecule which we have isolated for the first time from Indian basil, and with the use of extensive nuclear magnetic resonance spectroscopy, we come to the structures, and we found that it belongs to the phenyl propanoid class of compounds. Apart from that, if I want to summarize the basil in nutshell, because we have a big program on basil and its varietal development here in CMAP. So we have number of varieties marked by unique presence of compounds. If you see, we have a basil which can give you citrus kind of aroma. We have basil which can give you floral kind of aromas, and we have the basil which can give you beetle kind of taste. I mean, beetle leaf taste. So this sort of variation exists in osimum varieties. So in regular basis, our institute have released some of the potential lines for their mass cultivation. And now I'm coming to the point why the authenticity assessment is the need of R, because strict regulations on producing natural chemicals drive the production of the synthetic variants. Economically motivated adulteration is a serious concern because this essential oil market business is growing day by day. There are various routes for synthetic substitutes like citral, like menthol, like linalool. They can be produced by natural or by synthetic ways, and we have plenty of chemicals which are available at low price if synthetic production are concerned. Such supplies makes the synthetic option is more viable because of these. The low rate chemicals are the supply, and the price is very much comfortable for the producer or by the manufacturers. And one important thing is that fossil fuel byproducts, or what you can say fossil fuel based substitutes, they are one of the major starting material for production of these aroma chemicals, or for production of essential oils. So sometimes people, what they are doing nowadays, they are adding some high boilers like cooking oils. Like what you can take examples of vegetable oils. We have coconut oil, we have castor oils, or sometimes the petrochemical fractions are also being added into these oils just to make money out of that. So if you see, we have to now there is a need to regulate this the adulteration market, the business. So I will talk a little bit on three things: the gas chromatography based techniques, thermal gravimetry based techniques, and radiocarbon dating. By not taking much of your time, see what this mirror image is doing. Actually, we have done some enantiomeric differentiation studies. So basically, if you see, these methods basically differentiate the mirror images which are non-superimposable. So we have applied these methods in the essential oil which grow naturally, and what we found that they have a unique ability to produce a single image. Like I have done the study on menthol mint and in peppermint, and what I found that the minus enantiomers, like producing minus enantiomer dominating compounds are like limonene, menthone, menthol, and methyl acetates. So these are the molecules which basically, if you talk about the quality control of these molecules, you say they always comes as single molecules. So this is how our cyclodextrin, these texting methods help us in countering the frequent adulterations which is being done by the companies. But of course, there are limitations because they cannot give you conclusive information about origins. They can say that there is a single. They can say that the molecule is present as a single image, so you can assume it is as a natural. But sometimes it never happens because the synthetic route is equally producing the same kind of compounds. So what is the answer? We have the answer, that is, we are measuring the radioactive carbon-14 for those compounds, like those aroma molecules. If the same molecule is coming from synthetic route, how you will trace it that it is not it is not having modern carbons, the present day carbon, or the carbon which is coming through photosynthetic routes. So we have worked a lot on these molecules, and we found that C14 is a weak isotope of carbon, and it is present in all living organisms. The living carbon-based material, you talk about the plants, the animals, have a strong C14 signature because it has not yet undergone any significant radioactive decay. So if you want to quantify this carbon, you will see 100% bio-based value. But on the other hand, the fossil fuel derived material, any chemical which is coming through fossil fuel route, will devoid carbon-14. So what we are doing, we have skillfully used, we are just measuring the total C14 content in any material. So by doing that, you can estimate, you can easily narrow down, you can easily pinpoint this on the origin or the source of the material. So far we have done the studies on one of our flagship crop, that is menthol mint, and we have successfully shown that the menthol which is coming from plant is giving 100% bio-based value, and the menthol which is having synthetic origin or which is coming from petrochemical route having zero carbon or C14, or in nutshell, it doesn't have any C14 activity. So the entire research was further extended when we worked together with Bureau of Indian Standards, and in collaboration we have developed one standards on synthetic menthol, and the specification is already available at BI's website, where this standard talks about test methods for distinction, and it talks about HSN code of the product, and of course it talks about quality of natural vis-a-vis the synthetic. So we have done so far, we have addressed one thing, that if you want to target the product origin, carbon-14 is the best option. Now one more challenge is there: how you will differentiate a material which has been contaminated or misbranded with any vegetable oil or high boilers. So there are options. So what we have done, we have skillfully used thermogravimetric analysis. So this thermogravimetric analysis, basically we have a freedom to work beyond 1000 degrees centigrade. So means you can run your sample, you can test, you can analyze your sample up to 1000 degrees centigrade. So by doing that, we were able to distinguish the sample which is genuine or natural, or a sample which is adulterated with vegetable oils or high boilers. So there is a simple thumb rule: if you are getting single step degradation, so you have to assume it as a natural sample, a genuine sample. If you are getting multiple step of degradation, like more than one step, as you can see in lower left side of your chromatogram, which is showing 20% coconut oil in vetiver oil, you are seeing two different degradation steps. So each degradation steps is quantitatively gives you the approximate proportion of the material. Means if I say 79.7, means around, I'm talking about the 80% authentic sample of vetiver. means it has been adulterated with at least 20% of non-volatile material or the high boilers. So we have skillfully used this technique also, and all these methods has been well accepted by the industry. And in conclusion, if I summarize my findings, vetiver and patchouli oils possess diverse chemical structures, starting from monocyclic to tricyclic structures. Presence of catenane class in one of our variety is a unique feature so far we have done. Bisabolane class are characterized in patchouli as new entities. It's a very new class of components. And we have identified a new phenyl propanoids from the variety from the varietal development program that is called metausinol. We have seen a lot of alcohols, which was again a new feature for some of our interspecific osmum. And we have the mirror image studies which we have done using gas chromatography and the thermal gravimetric analysis methods for authentication are proven they are going to be a better milestone in coming futures. And as far as the C14 measurement is concerned, we have shown that measuring C14 content of any natural product is an accurate tool to determine if it is adulterated with any faucet derived materials. And at the last, I want to acknowledge Director CMAP and all project grants we got from DST, from CSIR, my research collaborator from CSR NBRI, from IUAC. They helped me in measuring the carbon 14 dating results. My students, of course, they all deserve this credit because their hard work ultimately brings all this matter, all these publication, and of course the Society for Plant Research and Professor Bhatnagar for giving me this opportunity to present my work before these August gatherings. And at last, all the audience, the online audience, my students, I'm again thankful to you. Thank you so much, sir. It's up to you, sir. Thank you.
Thank you very much, Professor Chanotia, for wonderful presentation and research work which you had been doing at CMAP.
Thank you, sir.
And I congratulate you once again for being nominated for Fellow of Society for Plant Research Award.
Thank you, sir. Thank you so much.
It's my pleasure. And for our audience, I should apprise that Professor Chanotia is on the editorial board panel of the Society for Plant Research and Vegetables.
Yes, sir.
And we expect much more involvement with the society and its journal.
Yes, sir. I'll be there.
Global upliftment.
I will
Thank you very
continue.
much.
Thank you,
Thank
sir.
you very much, Professor. Now I would request Dr. C. Gopalakrishnan. He is Professor, Department of Rice, Center for Plant Breeding and Genetics, TNAU, Coimbatore, Tamil Nadu. Dr. C. Gopalakrishnan, please. Are you here, Dr. Gopalakrishnan? Dr. Gopalakrishnan, are you here online? Dr. Amjad Hussaini, are you available? Dr. Amjad Masood Hussaini, your mic is mute.
Yes, sir.
So, Dr. Gopalakrishnan, we are moving forward for the next presentation. Till then, you kindly rectify the technical issue. Dr. Hussaini, can you give your presentation right now?
Yes, sir. Yes, sir. I will try. I will just.
Okay. Most welcome. At the outset, let me congratulate you for being conferred for this prestigious Fellow Award of Society for Plant Research. And I am sure that your presentation will provide fragrance of Kesar to everybody who are present over here.
Thank you, sir.
So kindly continue with your presentation.
1 second. I am just trying to see that.
If this is the presentation, it should be this one. Yeah. Sir, are you able to see the presentation?
Yeah, it is visible.
Okay.
Please continue.
Okay. On the outset, I would like to thank the Society for Plant Research for giving me this opportunity to present this work in this August gathering of eminent scientists. Keeping in view the expertise of all the scientists here, I have kept it as a bird's eye view presentation, and also try to stick to the time duration limits. So what actually I want to say here is like how the journey of science has taken me from from initial stages to now what I am trying to do. And what is common is that always trying to solve the issues which are pertaining to our society, wherein we need to address them as scientists. So here is it. I would like to welcome you all to this presentation. And this is when I started. Is the first slide visible, sir?
It is visible.
Okay, great. So I joined the University of Agricultural Sciences and Technology at Kashmir, that is SCOAST, in 2007, and was given the assignment of working in a research station, which is a rice research and regional station. That time it used to be called. Being a plant biotechnologist by training and having worked in JNU and Jamia Hamdard and Solon on biotechnology, it was a very new experience for me to get into the field experience of how it is done in conventional breeding. So I tried to fit in the system, and for what I first found was that there was a lot of biodiversity available in Kashmir, especially in the rice germplasm. And I tried to find out that what are they trying to do. We used to go in for conventional breeding and also tried to maintain the germplasm. So when we were giving this opportunity to maintain the germplasm, this was like okay, simply go in and from year to year maintain this in the field and try to record the observations. So in that, what I found was that there were certain genotypes which were promising, which were specialty rices, and there came a question in the university that why should we go in for research in rice when we are getting rice amply from Punjab. So that was a question mark on our own existence. And I told them that, sir, let us focus on specialty rices, and which was taken up, and it was supported by the leadership that time. And what we did was we first tried to see the specialty rices in the form of aromatic rice and in the form of pigmented rice. So the first aspect was to document them for the biopiracy aspect of it, and characterize them in the field. And the next was to nutritionally characterize them, and also to first purify them. So here in this slide, I'm showing that there was a concept of participatory varietal selection, PVS, as participatory varietal breeding, wherein we took the the germplasm at advanced stages to the field of the farmer, and then would see that if it is responding well in their condition, in the resource conditions being provided in the farmer's field, not in the resource efficient conditions of our own farms. So this was one of the aspects that we started to work on. And as you know, the agricultural biodiversity is very important in shaping the cultural diversity also. Like we have certain habits of food habits which are shaped by what kind of crops we have. So here we had this mushk butchi.
Mushkabudji is a rice genotype of short and bold seed type, and it is from Japonica line. Unlike the basmati types, it is short and bold, and the aroma retention is much more. So first aspect what we worked on was to purify this variety. There were many claims, it was as a farmers used to grow it, and then everybody would say that this is Mushkabudji, while as there were differences in morphology as well as in aroma content and as well as disease reaction aspects. So what we started under the leadership of our associate director and myself, being the junior scientist at that time, we first evaluated these and purified Mushkabudji on a single plant selection basis, and then checked for the preferred traits, especially aroma and disease reaction. And then this was taken up as a public-private partnership enterprise with Sarveshwar Basmati, a company from Jammu, and for commercializing it, and to give support to the local farmers who were almost losing interest in growing this crop. So this endeavor then was supported by ICR also by giving us a Genome Savior Award, and it was a big boost. And that is now you can see Mushkabudji in the farmers field being grown, and it is also being supported by the university. So this was the beginning of the career from the purification of the varieties, simple purification, and then I was moved to the Center for Plant Biotechnology. Our university decided that we have to start a biotechnology department, and we first made a very small beginning from a center. Since my background was from rice, so I took some of the varieties. All these are the varieties which we took, and my first student did the nutritional characterization under in our newly established lab. We tried to find out the here, if you can see, there is ZAG 1, 2, 14, ZAG. All these are actually these rice genotypes which are having pigmented nature. So what we try to see that if these are better nutritionally and address the issues of the malnutrition in our population. So this was the first student who carried out this work, phenols and carotenoids and anthocyanin and protein starch, amylose and fiber content. She did all these 19 genotypes, and then also tried to find out the SSR based differences, if there are differences in the germplasm. What I had anticipated was that there wouldn't be much of the difference because this was very similar, ZAG 1, 2, and they were from adjoining areas, not too far. But to our surprise, we could see, even using a very meager, only 24 SSR markers, we could see the differences among us these genotypes, which actually, at that time, people thought that, oh, there is some error, and the student was also confused that what to do and all that. I told her that, no, you have to, you just come up honestly with whatever you have done. And then what came to our mind was actually it is because of the very, in hill ecology, you have a lot of difference within a very small area. So that hill ecology may have actually contributed to having these diversity, this much of diversity, even among this very seemingly related genotypes. This was the first publication from this work from my first student. That's why I have highlighted here. Physiology and Molecular Biology of Plants was the journal. So this was very encouraging for the student, and for the department, it was also like, okay, he has set a standard for other master student. This was a master student to follow. Then came another student, the second in the row, and since I was working on Mushkabudji, I asked her that let us try to see if the mushkabuchi has the same, molecular mechanism of aroma, which is there in the Pusa Sugand and Basmati genotypes. So here what we tried to see was it was already documented that there is a BADH2 gene, which is very important in stress resistance. There is a mutation in this gene, and that causes the production of aroma. So what our hypothesis was that let us first see that there is one exon 7. If that genotype has got that in our genotypes, that mutation is present, or is it dissimilar in some other way, or may not be, it is not in that very target region. So here we can see, we putting in all the pods ourselves, and the student and myself, and there was the department was very in fancy, and nobody was there to help us. These are the genotypes, 8 of them, which we actually selected, and then we went in and checked for this deletion in exon 7, and we found that there was a 8 base pair deletion in the aromatic genotypes, which was consistent with the Pusa Sugand variety. We checked it for the Pusa Sugand 3 also, which was as a check, and the similar was in the Mushkabuchi. So we concluded in that sense that no, there is this seems to be conserved, and it is even conserved before its evolutionary very, very important deletion, and it is common among the indica as well as the japonica types. And also we tried to see if we can give breeders something which is useful to them, and then we validated BADX7, which is a marker, and that marker also gave us the intended amplicon size, which was 95 in case of aromatic ones because of the deletion, and in the normal ones it was 103 or 105 base pair. So that validation was also done. This work here is from Deepika. She came as a first PhD scholar, and then by that time I thought that okay, we also need to move towards cold tolerance. We have a lot of cold tolerant germplasm available in Kashmir, and we assume that all of that is cold tolerant, but under controlled conditions we had not checked that. So it was a DBT funded project, and therein we had a walk-in growth chamber wherein we could create these conditions under controlled conditions, and impose the cold tolerance, impose this cold shocks, and we tested, and we saw that this upregulation of these transcription factors was there, and this actually modulated the ROS levels in a time-dependent manner, and this was kind of these 3 transcription factors were playing a key role in imparting cold tolerance amongst the genotypes. I have skipped here, the screening part and all. There was a screening of around 100 genotypes, and then out of them we categorized the very tolerant, the most tolerant, and the less tolerant, and the susceptible ones, and then we compared the tolerant with the susceptible ones, and then we came up with this, that these MIP4, DREP1 and AP37 transcription factors, even though they are 2 of them are implicated in stress, AP37 is generally for developmental stages in functional role, but here we could see, and that is, this paper is now published. Another paper, then the nutritional profiles and fertilizer responsiveness of 50 genotypes was also seen. Among us, the promising genotypes of cold tolerant germplasm, so that the breeders can make use of this germplasm for based on their nutritional content and fertilizer responsiveness, apart from being cold tolerant. So we could see that yes, trait-specific markers were used in this, and they were successful in giving this presentation. And now coming to what you said, the saffron tinge to the presentation. Saffron, as you know, is a very important crop and niche crop in Kashmir, and it is the basic yeah. So this is about saffron. In saffron, you have many ways of improving saffron. What we did, I will just come. We first started that let us see if there is a difference between the saffron. There is a lot of saffron being grown for a lot of time, and is there some genetic variation between the clones? So we started with this, and then there were three promising clones which were there, and one of three of them were having tetrafed stigmas instead of the trifid ones, and but although there was a difference between the crocin saffronal and picrocrocin content between these clones, there was not much appreciable difference in the genetic background. So this is actually saffron is actually a monophyletic sorry, are you able to see here? Yeah. So it is so here, if you see, it is actually not having much of the appreciable difference. This is because it has been in monomorphic species. Is it able to move? It is hand.
Yeah, it is moving.
It is moving, sir.
Yeah.
Okay. But it is not moving from my end here.
You are at 14th slide now.
Okay.
And it is already there.
So, so this study actually showed that the amplification profile of the 31 saffron clones, there was not much difference in the saffron clones, although there is difference in the nutritional in the bioactive compounds in them. Somehow it has got hand. Yeah, it is moved. So apart from going for this thing that there is not much difference, then how do we improve? One aspect is through biotechnological approaches, and for that you need to have a protocol for regeneration. So before regeneration is to be started, why not to go in for micropropagation of saffron? Apart from improvement, it also can help in developing comlets for the mass multiplication. So this was the work which one of my PhD scholars did, and we developed an in vitro comlet production protocol for saffron, and wherein we got some success in getting these microcombs, and the microcombs of around 2.5 gram size using TDZ was considered to be a new by this regulator, growth regulator, and that had helped us to get this. Otherwise, it was a very recalcitrant type of plant wherein we could not get much. So also, we tried to find out that if there are any microbes associated with saffron, then we can use those microbes, multiply them, and then try to see if they can improve the size of the combs and also multiplication. So 6 isolates of rhizobacteria were identified by the student, and these are shown here, and each of them had some specific property. For example, Bacillus subtilis was the best strain for IA production, and Pseudomonas was for phosphate solubilization, and Cedrophore production, Pseudomonas was again the best one, followed by Bacillus subtilis and all. So here you can see that the co-cultivation of these PGPRs was also done in vitro to try to see the effect on the growth of these corms, microcorms, for organic saffron production. So, but by that time, I was convinced almost that, you know, the microbes which are isolating from these very environments are not giving us, you know, those desirable results. And also that if there are commercially available ones, why not to test them in our crop and see, give the farmer the best ones and recommend to them those commercially available ones. So here actually this slide has come up. This was a tissue culture. In the meanwhile, a student came and we started to work because of her being an background from forestry. Chinar is again a crop, is again a plant of very significant importance in our place. So we developed for the first time the regeneration protocol for the tissue culture protocol, multiplication, micropropagation protocol for Chinar, Vasia. So in the meanwhile, then I went to Oxford, and this was the work which was done there. We got a breakthrough, you know, paper also. This technology was developed there. This is called as the activity-based protein profiling, wherein you know, you can actually see the active proteins when they are in the active state in the cell. As you know that you know, the genome will give you an idea, but actually the functional protein, there are most of the post-translational modifications and transcriptional, during that transcriptional modifications are also there, and all that. And then you need to come up with some technology which actually gives you a snapshot of what is happening at the proteome level when it is active. So this technology is very, very novel, advanced technology being used in Oxford, and I was fortunate to get some breakthrough in it, and I used it in saffron. Here I have shown only that part which we used in saffron. Here, if you see that there were 4 stages in saffron stigma development, and we used the probes, different probes, which could target some of the alpha glycosidases, and then we could see that there is a prominent increase in the concentration of some, and there is a prominent decrease in the concentration of others. And then we could also see the active proteases after pull down and trypsin analysis and MS analysis. Those are given on the right side. Actually, I'm able to see only half of my slides. So anyway, then we also tried to see the, you know, if we can multiplex these probes with some other probes and see if these can help us in corm rot identification, which are the basic enzymes which are being upregulated or downregulated during this corm rot. So you can see here, if you the left is the mock ones, and then you have iridia. There are 4 specific these 5 specific strains of Fusarium oxysporium which were used, and try to see with each probe the changes in the cell, corm cells, what is happening. So if you can see there you can see in the bands, complete bands, full bands, 2 bands on the left hand side, on the right hand side also complete 2 bands, while as in the middle you see in the JJP 383 there are some novel bands appearing while some are disappearing, and similar is the case with you know, serine hydrolase probe. And there is in then overlay of these, and you can see them in different fluorescent colors, and see these protein gels are in the lower side. So coming back from Oxford, then I thought that you know, we got a project from NMHS, and try to see that, okay, if we can have this saffron in the non-traditional areas of Kashmir, because the traditional area of saffron is under multiple kind of facing issues there, owing to urbanization, owing to factory, and owing to the market prices of the land and all that stuff. So this study here we conducted in 18 different sites across 16 different districts of Jammu and Kashmir. And we also try to see that if we can have saffron in all these regions. These were ecophysiologically similar areas to the area which is the traditional area in the Pampur. So apart from that, we also try to popularize saffron in the among the women as saffron kitchen gardens, because saffron is having multiple medicinal benefits, and it is also a cuisine used in the cuisines of Kashmir. So we thought that, okay, let us also popularize it as a saffron kitchen garden. So this was very much appreciated as an organic kitchen garden, and in the non-traditional areas, we got some promising results. And especially in this, when I see these small children and the background of these farmers, which were from extremely poor backgrounds in far-flung areas in Jammu and Udhampur region, and in Kashmir, Kupwara, border areas and all. I was feeling that, okay, if our intervention is able to help them in their livelihood, and if it develops into an enterprise from them, nothing like that. So this was for the extension of this program. And then this is a this slide shows the foldoscope. Foldoscope came up as an invention from an Indian scientist in Stanford University, and DBT invited some proposals to use it as a frugal science tool for popularizing science among children, basically. And it is a low-cost device, 500, 400 rupees. So we were the first actually to see that, try to see if we can use it for detection of fake saffron. So it gets connected to a mobile phone, and it amplifies the pictures. So what we did here was we tried to see get all the fake genotype, for all the fake saffron samples from the market and the genuine ones, and we standardized the technique. And then we found that, okay, we have some visual markers in the genuine ones. Here, if you can see in the poster, having these finger-like projections towards the end. These are papilla. These are present only in the genuine saffron, while as in the fake ones, this is not present. So we developed a poster, and we tried to popularize it and gave some lectures in the schools, and also spread awareness among the target society. This is the basic difference is here that this is customer-centric approach, that the customer can use himself on a mobile phone and compare it with the poster. So then we thought that, okay, the poster may not be available to everyone and may not be able to understand. Then we also came up with an AI-based tool, which I am going to discuss here. Actually, I see another, because in the meanwhile, there was this COVID pandemic era, and everyone was at home. And what I tried to see was we didn't have access to the labs, wet labs. So we tried to find out that if there is some kind of a possibility of giving the scientific community a lead that saffron can also perhaps be used. So for mitigation of the post-COVID symptoms, post-COVID, traumatic, those disorders, stress disorders, and also in minimization of the direct effect of this. One of my colleagues, he helped in the bioinformatics part of it and tried to see the difference between the binding potential of the bioactive compounds, which I gave him the structure of, and with ACE2 receptor. And then we came up with this, that it may perhaps be a useful tool, and it should be further investigated in wet lab by this. So this was intervening in 2020, 2019, when the COVID pandemic was there, this study. So what I was discussing was that, we had developed this poster, and then we came up with this thing that why not to have some AI-based tool as an app to check. Anybody can check it using an AI based app. So you can download an app which is called the Saffron Rapid Test app, available on Google Play Store, and then upload a photograph from the sample, and then you can get it whether it is fake or whether it is genuine. So this is one thing. This was another work, because we are working also on tissue culture. Gladiolus red majesty is one of the floriculture crops, which is important, and we developed this protocol here, which is published in Vegetos itself. So one of the students came up with this micro. Then coming back to saffron, here we have now come up with this thing that microbial consortium, which generally we use that the term that any organic amendment can be beneficial to any crop, and we should use it. We checked many of the microbes and try to see that if the effect is equal, or is it different, or is it specific. So we came up with this thing that it is crop specific microbial consortium, and now we are going to upscale it and going for some startup as a student. The student will go for that. So here this microbial consortium is very useful in enhancing the column size as well as enhancing the quality of the saffron stigma. Column size, we use a different one, and for that we use another consortium. So then you have, this is the first report. Very recently, one of my PhD scholars came up that Fusarium acuminatum is a causal agent of corm rot in Kashmir. And we have identified it using polyphasic approach, integrating the markers and biochemistry and all. And then now what Mushkabuji is again coming back here in this slide, because my student Komal is taking up the CRISPR-Cas based genome editing. This Mushkabuji has a problem of blast tolerance. So it is very susceptible to blast. And if we have to engineer it, now that these CRISPR-Cas is coming up as a big, in a big way, and is acceptable also in India. We have now rice also in this. So we had started this program 2 years back, and we are going to develop CRISPR-Cas based genome edited Mushkabuji rice for blast tolerance. Then this is what I leave with. What difference do I make in my life? Is not that important. It may give us money and fame and scientific impact and motivation, but what difference do I make through my life is very important. That is, it should be meaningful to society, and scientific impact should be there, and it should create happiness and satisfaction. And my belief is that nobody remembers you for your contribution to you. They remember you for your contribution to the society, to them. So this was the whole premise of having all this collection of this. And this, I am thankful to the press and media for highlighting some of the work which we have done, especially most of it is pertaining to saffron. Here you can see saffron revival in Kashmir and Oxford work and being acknowledged, and then this app and developing of the calendars for spreading awareness among us people. common masses about saffron conservation and all. So in the end, I would like to thank all the former team members of my work. Each one has contributed to his or her maximum capacity. It is actually through teamwork that we come up with something which is useful to the society. It is not that you can individually do everything. So these are our hands. They are my hands. They from because of which we have accomplished some things here. And this I would also acknowledge the work, the support given by the funding agencies for carrying out all this work. And most importantly, I would thank you personally, the Professor Bhatnagar, Professor D.K. Srivastava, all the team behind the Society for Plant Research, who are doing excellent work in societal biotechnology, spreading good information and encouraging scientists to work positively and bringing in positive vibes in the system, in the scientific endeavor. With all these words, thank you very much all for having patiently listened to this talk. And I tried to be very brief. And if I have passed the time limit, I'm sorry for that. I couldn't tell you all the details. Most of this work is published, and this can be accessed, and anybody can also request. I can share freely all the published work with them. Thank you very much. Thank you for giving us this fellowship award also. This is certainly an encouragement, and certainly we will also try to actively engage with Vegetos, with the society, try to take it to the highest level under your able guidance and with the help of all the colleagues there. Thank you, sir.
Thank you, Professor Amjad, for wonderful presentation. I know that you have got enough bulk of information to share, but we always run short of time when such webinars are organized. So, but sometimes we will certainly organize the webinars taking one or two scientists on the thematic area, so that we shall be able to enjoy the detailed information, whatever your lab and lab mates are doing under your supervision. Thank you very much, and congratulations once again for being conferred this award.
Thank you.
Thank you.
Yes,
now, Dr. Gopalakrishnan, are you able to speak something about your work? I think he informed that he is not having the network over there. So I think one more award was there by Dr. Jeffrey from USA, but I understand that the time zone difference is such that it is not worthwhile to ask him to make presentation in the midnight almost. So thank you all very much. And this was the mandate while conferring awards for last 2-3 years, that simply evaluating the nominations received and the biodata CV received. If we make a judgment, then it will not be fair on our part. Let us have an interaction between those scientists who have been conferred the award, so that there should be a close bonding between the awardees and the society as well. So this was the motive. And therefore, our Executive Council has made it a mandate for all the awards that we shall have a presentation. Of course, it is a time-consuming process, but when you want to interact, when you want to share science with others, ultimately time should have no limit. So we should always not bother about it. Likewise, you will see that the audience for scientific gatherings are very less. They are reducing. If you organize a cultural night, you will find the hall full. So this has become the scenario. For scientific listening, you will find persons who are really interested in science, and who want to contribute to the society, as Dr. Hamzat said and Dr. Sanket said, that we have to contribute to the society. That is our prime motive. As Society for Plant Research, when I established it in 1988, 36 years back, I am now crossing 70. I have now crossed almost 70. So when 36 years back we founded this society, we never thought that we shall be having so much of affection and love from the scientific community that we will reach to this platform. So that was a dream, but that was a dream. And like Arjun, we were having a target to achieve. And I am very happy to confess that we are moving towards the achievement of the target. With the help of all fellow scientists, esteemed workers, young faculties, fraternities, and supporters. So this is very important. When you start anything, starting a journal or starting a society might be an easy task. But to pull it on for 36 years or to pull it on for 40, 50 years, that is a hard task, at least for a person like me. But with the help of everyone, I could achieve it anyway. You know that its Silver Jubilee volume was released in NAS Complex, New Delhi, by the then Director General, Professor S. Ayyappan, who is unfortunately no more now. So, and that was the Conference of Vice Chancellors and Directors, where this 25th volume, Silver Jubilee volume of Vegetos was released. So, right from that moment, we started thinking that with the help of everyone, we can do something great. And Vegetos, I suppose, as Dr. Hamzat said, and he had been guiding me and supporting me at various steps, that the journal is the most loved one journal as far as biological sciences or plant sciences are concerned. So I expect that all of our fellow awardees and the attendees who are present over here this afternoon, they will motivate and will tell the objective of the society, the mandate of the society, and the journal and its review policies and the quality which we had been publishing. We had been trying. I am not saying that we are publishing 100% quality papers. Sometimes there is a skip from the basic flaws. And you know that from Elsevier, a large number of papers have been retracted. And there is a continuous process of retraction of papers from the journals, very established journals. And luckily, Vegetos has not come to that threshold. This is because of our editorial board, because of our well-wishers, scientific community who are associated to us, and the reviewers. Therefore, we are able to achieve this goal. And this is not the limit, this is the beginning. And we have to go much far away. Of course, the founders may remain or may not. But the things are in the hands of younger generation. They will have to see that how the legacy can be maintained, how the journal can reach to the topmost position in the world, in the plant biological sciences or biotechnological sciences. So this is all up to the younger generation. So I thank you very much, and the forum is open for discussion just for 15 minutes because we are running short of time. Some persons wanted to ask to the awardees, so they can make questions one by one, please. Thank you. Yes, Dr. Vasudha.
Yes,
please
actually,
speak.
I wanted to listen to the lecture on seed priming, but I could not listen to it today. Is it possible to reschedule or something like that?
No, rescheduling is not possible, but we will try to.
No, I
produce,
think the
it on
lecture
YouTube.
was to be, yeah, the lecture was supposed to be scheduled today in number 3, but it was not held at that time, and later on also it was not held.
Dr. Anayatullah, he was away and he was not having the network over there. So we made a request to him again and again, but perhaps he had to leave his station for some important cause. So he could not be connected. But we will reschedule those 2-3 lectures at an appropriate time. It will be notified on our website. It will be notified. And all the lectures, if our awardees permit, then all the lectures will come on the YouTube and they can be accessed through our website also, vegitosindia.org. So that more people who could not attend this webinar, they may take benefit of the lectures. So,
Thank you, sir. Thank you.
and I think, is there any more query? Otherwise, we can take leave. Dr. Marina, do you have queries?
No, sir.
Dr. Vineet Soni?
Sir, namaskar. Good afternoon, sir.
Good afternoon.
And thank you very much for organizing the wonderful session. And really, it is a big opportunity to hear all the speakers, all the SPM fellows, sir. That is and I hats off you for your contribution. And presently, even after from longer period, you are running an excellent esteemed journal, sir. So this is really a big work in the field of science from your side, sir.
God has provided me some strength so that I am able to serve science. That is all.
Really.
And the cooperation of you all. I will ask my office to share a small 12-minute documentary to all of our awardees so that they can know that how this journey could be performed for 36 years. It's a small documentary that will be shared and it is available on the YouTube as well. I would request all our fellow awardees to kindly go through that thing so that at least they will be able to see that how did it originated and how could it reach to this extent.
Thank you very much, sir.
Thank you all. So if there are no questions, then I should express my greetings, congratulations, and thank to all our esteemed scientists who spared a lot of their valuable time to be present and to apprise of their contribution. Of course, these awards, they are the motivating force, and these are just beginning if we talk about the career, because I have completed my 41 years of career in teaching and research. So for younger generation, it may be a little motivation that if they continue to work, then their work, there is no dearth of recognition. At least this society is always all set to recognize the contributions at any level, whatever it is possible. So I congratulate you all once again. Professor Vanna Bose, our president, is indisposed for due to some vaccination or something. So she conveyed me that you kindly express greetings and congratulations on my behalf to all SPR awardees. And thank you all very much. See you once again. Keep contacting and keep connecting. Thank you.